where we are concerned today with chemosensory biology and the interface with immunology. So immunology and chemosensory biology share some common challenges, which is the recognition of what is foreign and bad and bitter and pathogenic from what is good. And so one of the central tenets of what we're doing today is that by understanding these two systems and sharing these common problems that both disciplines, when studied together, we learn more when we know more about the intersection of chemosensory biology and immunology. So I also just wanted to take an opportunity to say thank you to all of our speakers and to those of you who are in attendance. And also I wanted to just give a shout out to Katie Luong, who was our administrative coordinator and herded the cats on this particular project. And I also wanted to thank my co-chair, Kevin Foskett, who will be leading the way here. So our plan is that we'll have Kevin give us an orientation to intracellular taste signaling. We'll have a keynote address by Noam Cohen from the University of Pennsylvania, who's also an adjunct faculty member at Monell. And then we'll have a series of short talks, and we'll be capped off by Dr. Nancy Rawson, who will give us a keynote address about sort of the mutuality between Penn and the University of Pennsylvania and the intersection of these disciplines. So I know that Kevin is eager to tell you, to give you an orientation about intracellular signaling. So I will turn it over to him. Kevin Foskett.
Great, thank you, Danny. Let me try to share my screen and let's try this one. Can you see it?
Yes, we can see it.
And is the forwarding working? No. Let me do it a different way. Sorry.
No worries.
Try the forward arrow, Kevin.
No, I told you I was going to take up all of your time.
Yeah, but I was hoping you'd be talking during this, Kevin.
Can you see a slide?
I can.
I can't seem to forward any. There we go.
There you go.
Okay. Yeah. Okay. So very sorry. Thank you very much, Danny, for the introduction, for the opportunity. I'm going to give a brief overview of taste in the periphery, and I've indicated two different logos on here. I'm at Penn, but in full disclosure, I'm also on the board of trustees at Monell, and why I have an affiliation with Monell will become clear during my presentation. So as Danny pointed out, the sense of taste allows humans and animals to test food before they eat it so that they can distinguish foods with nutritional value, something sweet or umami, for example, from something potentially harmful or toxic, usually with an aversive taste like sour or bitter. And in the oral cavity, in the periphery, and we won't be talking too much about the periphery today because we'll see that in many of the talks, the taste system is actually widely distributed throughout the body, but in the oral cavity, tastins or chemicals in the food are detected by specific receptors expressed in taste cells that are clustered in taste buds, onion-shaped structures that are found in the tongue and the soft palate in the oral cavity. So taste buds are the basic unit for processing taste information in the periphery. Each taste bud comprises a heterogeneous population of anywhere from 50 to 100 tightly packed cells, with each cell representing one of 5 basic taste qualities: sweet, bitter, umami, sour, and salt. And importantly, and very interestingly, taste cells undergo constant turnover throughout life, and they're replenished then by adult taste stem or progenitor cells in or around the taste buds. Next slide, if I can get there. Do this, okay. So the mammalian taste system is a prominent example of a major sensory system that's hardwired to trigger predetermined actions and behaviors such as innate attraction, consumption of sweet compounds, for example, and aversion to bitters. And these activated taste receptor cells on the tongue and the palate epithelium transfer their signals in a label line system. That is to say that each gustatory neuron is dedicated to each taste bud cell, transmitting that information about the quality of the taste ultimately to the cortex in the brain. And as I just mentioned, because these taste cells are turning over all the time, it's a very interesting cell biological problem for which there's a little information, but not much, about how this label line is maintained in the face of ongoing cell turnover in the taste bud. So, sorry, this is the way I have to forward these slides. So as I noted, each cell in a taste bud senses one of 5 basic qualities: sweet, bitter, umami, sour, and sodium. And these cells can be distinguished in part by morphological criteria, but also by their gene expression profiles. The interaction of tastins in the oral cavity with the taste cells is mediated by receptors in the apical membranes that protrude into the oral cavity. And these receptors fall into 2 basic classes. G protein couple receptors bind to sweet and umami and bitter tastins. Cells that respond to sweet use a heterodimeric GPCRs comprised of T1R3 with TR2 or 1, whereas bitter taste GPCRs are of a different class. They're believed to be monomeric, and there are over 40 with splice variants, as I understand it. In addition to GPCRs that mediate sweet, bitter, and umami taste, the taste of appetitive salt and sour taste is mediated by ion channels. So salt is sensed by the epithelial sodium channel ENaC, which is very important in the kidney and the lung and the gut for sodium transport, but it's important, as we'll see, in the taste system for perceiving salt. And a recently discovered ion channel called OTOP1 that mediates sour taste. This is a sodium channel, and this is a proton channel. So the signal transduction for sour transduction in so-called type 3 cells that express OTOP1 is mediated by a change in intracellular pH, an acidification of pH by a sour substance, either by a weak acid diffusion into the cytoplasm that acidifies the cytoplasm, or proton influx through the OTOP1 ion channel. This acidification inhibits constitutively active potassium channel, causing it to close. And when it does, that depolarizes the plasma membrane, activating voltage-gated sodium channels, which generate action potentials. And these action potentials trigger the activation of voltage-gated calcium channels, enabling calcium influx from the extracellular medium into the cytoplasm, and this triggers synaptic vesicle release of neurotransmitter in really a classic model of excitation secretion coupling. Before I talk about how so-called type 2 cells mediate perception of sweet, bitter, and umami, I'd like to take a little bit of a shortcut to explain how we got involved with Monell. And that actually came from a very unrelated project shown here in this publication from Cells some time ago, in which, through a different collaboration, Horia Vai in my laboratory, and then subsequently Zhangming Ma, established that a newly described gene, which has 5 different paralogs, which was referred to as CALM1, is a novel voltage-gated ion channel. Here we're showing a series of currents through this ion channel in response to depolarization of the cell membrane in a voltage clamp experiment. What we subsequently showed, using a variety of techniques, and this is work carried out by Adam Siebert as a graduate student in my laboratory, using fluorescent dye uptake methods, or shown here some electrophysiological approaches, was that the pore, the pathway for ions to actually move through CALM1 was extraordinarily wide. We estimated that the diameter of the pore was about 14 angstroms. That's remarkable for an ion channel. Subsequently, and quite recently, cryo-electron microscopy structures of CALM1 have been solved. And you can see in these structures, the pore diameter is very close to what we estimated in our own studies, 14 to 15 microns in diameter. What does that mean? Well, for us, that was really an aha moment. It's one of these very special moments in science that makes science so exciting, when things just kind of become obvious and exciting. And why was that? Well, because we knew that in type 2 cells that respond through GPCRs that I just mentioned to sweet, bitter, and umami tastings, that they, even though they each express their own cohort of either sweet receptors or bitter receptors or umami receptors, they all possess a canonical signal transduction cascade downstream of those receptors, involving an activation of a heterotrimeric G protein that activates a phospholipase C. In taste cells, it seems to be specifically beta 2, and this hydrolyzes phosphatidylinositol bisphosphate in the plasma membrane to generate a soluble second messenger IP3, which binds specifically to the type 3 isoform of the calcium release channel, the IP3 receptor localized in the endoplasmic reticulum, which releases calcium into the cytoplasm. And this calcium stimulates an ion channel of the TRPM classification, specifically TRPM5, as a calcium-sensitive ion channel, which is permeable to sodium. This depolarizes the cell membrane, activates sodium channels and sodium action potentials, which leads to the release of neurotransmitter. And previous studies that suggested that ATP must be the neurotransmitter, but the remarkable feature of type 2 cells is they lack classical synapses. They lack the synapses that we saw in the type 3 cells in response to, that respond to sour tastings. And so the notion was that perhaps some other type of mechanism, maybe an ion channel mechanism, enabled the release of ATP. Well, I previously worked with ATP, and I know how big ATP is, and the pore diameter that we discover for CALM1 is big enough to enable the permeation of ATP. So we looked into that, and in a really, it was so good for us that Monell is just 2 blocks from us, and we've had a really terrific collaboration with Mike Tordoff and Ichiro Matsumoto, who have enabled us to establish that in fact, CALM1, in fact, it's a CALM1-3 heterooligomeric ion channel is responsible for the release of ATP. And in the absence of CALM1 or CALM3, mice cannot taste sweet, bitter, or umami because they're unable to release neurotransmitter through this mechanism. Subsequently, Aki Taruno, who led some of that work in my lab in his own laboratory in Kyoto, has established the identity of a new cell called the sodium cell. And this is work that was complemented by recent studies from Ichiro Matsumoto at Monell, demonstrating a very similar but unique type of signal transduction mechanism for cells to respond, for these cells which are relatives of type 2 cells to respond to sodium chloride. And here, sodium permeation through the ENaC ion channel depolarizes the cell to trigger the sodium action potentials, which then lead to the release of ATP, again through these CALM1-3 ion channels that we had discovered for sweet, bitter, and umami taste. And so this is a little schematic showing, summarizing the signal transduction mechanisms for sweet, bitter, and umami transduction in type 2 cells, and a very similar mechanism that has no reliance on intracellular calcium, which is mediated purely as an electrical signal that Aki Taruno discovered. And this new type of synapse mediated by an ion channel, Aki has referred to as a channel synapse. And in my last slide, I'd just like to show why that's very special. So although these cells lack traditional types of synapses and are unable to package ATP or neurotransmitters into secretory vesicles, they nevertheless localize the CALM1, the release channel, to very discrete puncta in immediate proximity to the gustatory nerves. The cell biology that enables CALM1 to know precisely where the nerve is, or the nerve to know precisely where CALM1 is, and what localizes it to this puncta are studies that await research. So just to acknowledge the people in my lab, especially Zhangming Ma and Aki Taruno, and the wonderful collaboration that we've had that have made our studies possible. Particularly big shout out to Mike Tordoff and Ichiro Matsumoto. Now, just as a prelude then to end my talk, I've talked about the peripheral taste system, and what we're going to hear about today is how widely distributed the taste system is. And this is just a little summary slide that I picked up someplace demonstrating that taste receptors that I already alluded to that are present in taste bud cells are also distributed throughout the body. And in talks after mine, we're going to hear a lot more about that. So thank you very much. Happy to take any questions if there's any time.
Yes, so we have maybe just 1 minute if somebody wants to ask a question out loud, but mostly I would encourage you to put your questions to Kevin Foskett in the chat, and he will be able to answer those to you directly. So if we have anybody that wants to ask that one burning question, I'll take that as a questions to chat then. So that leaves me next to, of course, thank Kevin for that, for helping us really get a handle on intracellular and cell-to-cell communication and to bring this new information to the chemosensory biology community. And what's up next is, it's my pleasure to introduce Dr. Noam Cohen. He has both affiliations with the University of Pennsylvania, and also he's on the adjunct faculty at Monell Chemical Senses Center. Noam really is a real dynamo. There's no other way to say it. He's a very active clinician in otolaryngology, a very fine basic scientist, and he's really able to understand the commonalities between the perception of bitters and pathogens in the human nose. And so I will now leave Noam to tell you his story. Noam?
Okay, so can you guys see my screen?
We can see your screen and we can hear you.
Okay, very good. Well, thank you, Danny, and thank you, Kevin, for the introduction. I'm Noam Cohen. I'm one of the otolaryngologists at Penn, and within otolaryngology, I subspecialize in rhinology. And what I'm going to tell you about over the next half hour or so is the work that our division has been doing over the last 10 years, looking at the role of taste receptors as regulators of upper airway or sinonasal innate immunity. Now, on the clinical realm, I'm in the same boat as Kevin. I got to figure out how to advance. Okay. In the clinical realm, in the division of rhinology, the main disorder that we take care of is Rhino Sinusitis, which is a catch-all phrase for persistent inflammation or infection of the nose and paranasal sinuses. From a symptomatology standpoint, think about the worst cold that you've had, but those symptoms persist for months to years. So in your nose, loss of sense of smell, facial pressure, headache, inability to concentrate. So it's a big quality of life type of disease. It's relatively common. It's costly. It keeps us as surgeons relatively busy. And the fact that I really want to point out here is that it is the number one diagnosis for the prescription of antibiotics in the outpatient adult setting, 1 in 5 antibiotics. So it is a major contributor to the problem that we're having now with antibiotic resistance. And this is one of the facts that motivates a lot of the research that we're doing in our division to try to come up with alternative therapies for these patients with rhinosinusitis that would spare conventional antibiotics. From a quality of life standpoint, patients with chronic rhinosinusitis score worse than patients with COPD, CHF, and angina. Now, the management in a nutshell is relatively simplistic to start forward. We usually have our patients start irrigating out their nose with a neti pot to try to get rid of all those inflammatory and infectious mediators. In addition, some either local or systemic anti-inflammatories like steroids. And at least in our practice, we try to utilize culture-directed antibiotics if we see purulent secretions. We usually intervene like this for 4 to 6 weeks. And if the symptoms resolve, great. And if not, then we get a little bit more aggressive and we offer them surgery. And the type of surgery that we do for these patients is called functional endoscopic sinus surgery. It was first described by Dr. Kennedy in 1985. He was our former chair here at Penn. And in essence, what we do, this is a little schematic through the paranasal sinuses. So this would be the orbit, this is the roof of the mouth, and this is the nasal cavity. And in the non-operated cavity, what you see is a whole bunch of little bony partitions that basically trap the mucus. And if these sinuses are lined by mucosa, and if there's inflammation or infection, you get edema, that blocks off the outflow for the mucus. The mucus gets trapped, builds up pressure, driving the symptoms. And so what the surgery really is a marsupialization of that cavity, where we come in and we very elegantly or delicately remove all these little bony partitions so that we have a common cavity. The goal here is not to get too high into the brain or off to the side into the eye. But once that we do that, it allows the mucus to flow out even in the presence of inflammation. Now, we've been doing this surgery in our field for over 35 years, and overall, we have about a 75% success rate. So the other motivating fact driving a lot of the research in our division is trying to improve on that 75% success rate, be it with tailored surgical intervention or adjunct therapies to the surgery. And so I want to show you one of the patients in the 25% that did not do so well with our surgery. So this is a nasal endoscopy in the left nostril. This is the left inferior turbinate. This is a wide open maxillary sinus. The sphenoid sinus is here, and you can see it's a wide open cavity. But even in the absence of any anatomic obstruction, this sinus is not draining out. And you can see purulent secretions here. If we culture it and send it off to the microbiology lab, it comes back as Pseudomonas, and it's pan-sensitive. And I can put this patient on any one of these antibiotics, and while they're on those antibiotics, their symptoms resolve. But shortly after they come off those antibiotics, the symptoms come back. And as we go through this cycle over and over and over again, this infection now will become polymicrobial, and we'll start seeing resistances pop up in our sensitivities. So based on work that came out of Tom Finger's lab at the University of Colorado, and a lot of it was done by Marco Tizzano, who's now at Monell and will be speaking a little bit later on in the morning, and work coming out of Mike Welsh's lab at University of Iowa, we made the following observations. And when I say we, a lot of this work was done by Rob Lee, who is now an assistant professor here at Penn, but he was a postdoc in my lab doing this work. So what we observed based on this work was that the bitter taste receptor T2r38 is expressed in the human sinonasal epithelium, and its expression is enriched in the motile cilia. Again, based on the work that was coming out of these two labs, we also demonstrated that an endogenous ligand for T2r38 are quorum sensing molecules secreted by gram-negative organisms, such as acylhomoserine lactones. And when T2r38 is stimulated, you get a small rise in intracellular calcium, as Kevin demonstrated in the first talk. But in the ciliated cells, it drives activation of NOS, and you get rapid production of nitric oxide. In the airway, that NO has two physiologic consequences. It increases mucociliary clearance, and it diffuses out into the mucus and into the airway, where it can either be bactericidal or bacteriostatic, depending on what microorganism you're looking at. So to put it into a picture, bacteria come into your nose, they start secreting their quorum sensing molecules to communicate with other bacteria. This bitter taste receptor on the cilia detects it. You get a small rise in intracellular calcium, activating NOS. The NO diffuses out into the airway where it kills the bacteria, and you've increased mucociliary clearance so that you don't even have these dead bacteria around to be pro-inflammatory. Now, as Kevin alluded, there are many bitter taste receptors. In the human genome, there are approximately 25 genes encoding these bitter taste receptors. And as he also showed in his last slide, that over the last 10 years, it's been shown that their expression is not limited just to the tongue. So there are extra gustatory receptors in the respiratory mucosa, and the GI mucosa, and the GU, and the thymus, and Kevin had a lot more extensive list. One of the things that's unique about this subfamily of G protein coupled receptors is that there is tremendous genetic variability in these receptors, which then translates into a variability in individuals' ability to taste bitter compounds. And the best example of that is to the bitter compound phenylthiocarbamide. And so over the last 100 years, in thousands, tens of thousands of individuals, this following pattern has been established. And this is in the Caucasian population, that about 30% of Caucasians don't taste PTC at all, 20% detect it at incredibly low concentrations, and it's pretty repulsive, and 50% are somewhere in the middle. And based on the ability to taste PTC, in 2003, through positional cloning, the receptor was cloned, and the genetics behind that taste perception were identified. And so there were three polymorphisms within the gene that constitute a functional versus a non-functional receptor. If you have a proline, alanine, and valine at these three locations, that T2r38 detected PTC. If you had an alanine, valine, or isoleucine at those three locations, that receptor did not detect PTC. 85% of the time, those polymorphisms segregate together. And as I mentioned in the prior slide, in the Caucasian population, it's a balanced polymorphism. Nearly 50% of the alleles are alleles are non-functional, and 50% are functional. In Asians, it's 30-70, and in Africans, it's 10-90. So if you have two haplotypes, that generates basically three genotypes, homozygote functional, and this basically translates to super tasters for that molecule PTC. Again, in that context for PTC, homozygote non-functional, and they don't detect PTC at all, or heterozygotes. Now, the experimental paradigm that we use in our lab is the human air-liquid interface culture. And so, as I mentioned, we do a lot of surgery here at Penn and at the VA. We bring the tissue that is not needed for pathology over to our lab. We dissociate the cells up, and we plate them out on what's called a transwell. This is a plastic porous support that we've coated with either collagen or matrigel. You put your epithelial cells down on it, and you grow them for a couple days until they form a monolayer with tight junctions. Once those tight junctions are formed, we can aspirate the media from the apical compartment. And when we grow these cells in this air-liquid interface, it forces them to differentiate into a ciliated epithelium that recapitulates the respiratory epithelium. And this is what these cultures look like by scanning EM. Here you can see on a crosscut, that's that plastic support with the pores, a pseudostratified epithelium with the brush border. And on bright field microscopy, you can see the cilia beating. So we wanted to know whether the genetics of T2r38 that regulate the ability to taste PTC also translate into the ability of the receptor in the nose to detect those quorum sensing molecules. And so what we did is we grew our cultures from patients that we genotyped that were either homozygote functional for T2r38, so PAV, PAV, homozygote non-functional AVI, AVI, or PAV. Rob then loaded them up with an intracellular dye called DAF-FM, which fluoresces in the presence of intracellular NO. And then we stimulated them with increasing concentrations of the gram-negative quorum-sensing molecules. So this is C12 homoserine lactone, this is C4 homoserine lactone. And what you can see in the tracings is that there is both a concentration-dependent, but more importantly, a T2r38 genotype-dependent response to these quorum-sensing molecules, where cultures derived from patients that were homozygote functional for T2r38 at 10 micromolar already start generating a lot more intracellular NO compared to the heterozygotes or the non-functionals, and the same thing with C4HSL. And the other thing to point out is the x-axis here, which is time. So this is a relatively quick response. Within 5 minutes, you already get statistically significant differences in the amount of NO being generated if you are homozygote functional for T2r38 or if you're homozygote non-functional. Now, as I mentioned, the epithelial NO in the airway has two physiologic consequences. Through activation of protein kinase G, it increases ciliary beat frequency and mucociliary clearance. And then it also diffuses out into the mucus and into the airway, where depending again on the organism, it's either bactericidal or bacteriostatic. So we can take our cultures and we can overlay them with little fluorescent beads, and we can measure how far each bead travels for a given period of time, which is basically a proxy for mucociliary clearance. And so in this experiment, Rob took cultures from homozygote functional individuals or homozygote non-functional, measured how far each one of those beads traveled over 200 milliseconds. And what you can see is that basically at baseline, you get about the same amount of travel. We then took conditioned media from Pseudomonas that has all those quorum sensing molecules, and we put it on the top of our cells, and you can see how much further each one of those beads travels in a homozygote functional receptor compared to a homozygote non-functional. Graphically, that's represented here. You get a near doubling in mucociliary clearance in a homozygote functional receptor compared to a homozygote non-functional. And if we inhibit NOS with L-name, or if we quench all of the intracellular NO with CPTIO, we basically take a PAV culture, or homozygote functional, and convert it into the phenotype of a homozygote non-functional, and you barely see any acceleration. So that has to do with the T2r38 and the NO and mucociliary clearance. What about the antibacterial activity? And so in this experiment, we took cultures from our three different genotypes, homozygote functional, non-functional, heterozygote, infected it with Pseudomonas for 2 hours, let the bacteria proliferate, secrete all their quorum sensing molecules, stimulate the receptors here, release the NO. And after 2 hours, we collected the mucus with the bacteria, put it on a cover slip, and did a live dead stain. So anywhere you see a green dot, that's live bacteria, a red dot is dead bacteria. And here's the results of that experiment. Live channel, dead channel, and the merge. The columns are the different genotypes. So bacteria exposed to a homozygote functional, a PAV culture for 2 hours, the vast majority of the bacteria are dead, compared to the same amount of bacteria exposed either to the heterozygotes or the homozygote non-functionals, the vast majority of the bacteria are green. If we take the same amount of bacteria and we incubate them for 2 hours in saline, that's the ceiling of the experiment. Our positive control is the potent anti-pseudomonal antibiotic colistin, gives us about a 95% kill over 2 hours. And if you look at how well a homozygote functional ALI does compared to colistin, it's near comparable. Again, here are the heterozygotes and the non-functionals, and if we take nitric oxide out of the equation, we basically reverse that bactericidal activity. So does this have any clinical significance? And so we went back to all the patients that we were operating on, and we pulled their microbiology reports from the operating room, and we binned them into two different groups, either no growth normal respiratory flora, or patients that harbored gram negatives in their nose. We already had them genotyped for T2r38. And so again, I should also say that all of the genotyping that we did, this is in collaboration with Danny Reed at Monell. So she genotyped 3 other bitter taste receptors with nearly the same minor allele frequency as T2r38. And this is what we found. In the no growth normal respiratory flora, an equal distribution amongst our 3 different genotypes and throughout the other different receptors. But what was so striking was that in the patients that grew out gram negatives, we didn't find a single homozygote functional for T2r38 that harbored gram negatives in their nose. And this was a relatively small study. This was in 56 patients, and you can see what the p-value was. So again, a very narrow question, but a highly significant observation that T2r38 genotype and gram negative sinusitis. Now, I mentioned that one of the motivating factors behind the research that we're doing is to try to improve our surgical outcomes. And so we looked at patients that we were doing their initial sinus surgery at Penn. It was 123 patients. And in rhinology, we're not as advanced as, say, the cardiologists that have their echoes and their ejection fractions, or the pulmonary guys that have PFTs. In rhinology, we have a metric called the SNOT-22. This stands for the Sinonasal Outcome Test. It's a validated questionnaire of 22 items that assesses the function of the nose. And it goes through subdomains like nasal obstruction, olfaction, facial pressure, ability to concentrate, and sleep. The higher the score, the worse the function of the nose. And so to look at the effectiveness of our surgery, we look at the change in this SNOT-22 prior to surgery and then 6 months following surgery. The minimal impact in order for the patient to appreciate that the surgery did something for them is an improvement of 10 points. And so in general, in these 123 patients, we had a 25-point improvement. So for the most part, our patients are pretty happy with the surgery. If we look at this outcome measure in the context of the T2r38 genotype, not much to get excited by. Here are our homozygote functional with about a 30-point improvement after surgery versus our heterozygotes or our non-functionals with about a 24-point improvement. Maybe a trend that they're doing better, but nothing really to get too excited about. Now, one of the things that I've neglected to mention up to this point is that sinusitis comes in two flavors. Without nasal polyps, which is that first nasal endoscopy that I showed you, which is more of a purulent infectious process. It's a Th1-driven inflammation. And here you can see on the radiology, air fluid levels in the sinuses. You get a little bit of thickening of the bone, so an osteomyelitis or osteitis. Compared to the other broad classification, which is chronic sinusitis with nasal polyps. Endoscopically, this is very different. You see these big bags of fleshy-looking material in the nose. This is what the CT scans look like, a complete whiteout. And this is a predominantly eosinophilic or Th2 inflammatory cascade. So two very different processes, however, manifesting with similar manifestations or symptoms of nasal obstruction and facial pressure. If we look at our surgical outcomes and we divide our patients based on these broad inflammatory cascades, we see a difference. So in the type 2 inflammation or in the polyp patients, we get a good response, whether they're homozygote functional or homozygote non-functional after our surgery. So a 30-point improvement. Compared to our Th1 inflammation, where here we see a stark contrast. Our homozygote functionals do exceptionally well with our surgery, a nearly 40-point improvement in their quality of life symptoms. Compared to the heterozygotes or the non-functionals, they barely have a clinically meaningful improvement. And so I mentioned at the very beginning that this surgery is about 75% successful. If you look at this cohort of 123, 34 out of 123 is 27.6. So we think that we've at least identified a genetic biomarker that may help us explain which patients are not going to do so well with our surgery. And what we're now trying to figure out is, what do we do with these patients? Do we treat these patients as immunocompromised patients, kind of like a CF patient, and alter our surgery so that we, instead of having functional endoscopic surgery, we do gravity-dependent surgery? Or are there alternatives to the post-op management that we can try to make up for this immunodeficiency? Now, these observations have been confirmed in several other studies in Canada, in Poland, Australia, and in Charleston, South Carolina, confirming the role of T2r38 in this chronic sinusitis. There was one study out of Italy that refuted our findings, but that was predominantly a nasal polyp study. So I've focused so far on T2r38. What about the other taste receptors? So here's some physiologic evidence that there are others at play. Colchicine, which is used to treat gout, and dapsone, which is used to treat leprosy, both taste bitter, are specific agonists for T2r4. And so if we take our air-liquid interface cultures, we load them up with DAF-FM, we see a dose-dependent increase in intracellular NO to both colchicine and dapsone. If we do it in a T2r38 homozygote functional or a T2r38 homozygote non-functional, we get the same amount of NO, demonstrating that this NO response is being driven not through T2r38, but through another taste receptor. Here's some evidence that T2r16 is doing the same thing. So here's salicin, a specific agonist for T2r16. If we block the canonical signaling that Kevin was talking about with the phospholipase C beta 2 with U73122, we blunt that response, and here's the inactive enantiomer. So This demonstrates that there are multiple bitter taste receptors expressed in the sinonasal epithelium, and when stimulated by specific agonists, they all converge on the stimulation of nitric oxide. Here's a little bit more evidence demonstrating that pretty much all 25 bitter taste receptors are expressed in the human sinonasal epithelium at different levels. Now, I want to switch gears a little bit for the second half of the talk and show you a different response. And in this experiment, Rob loaded up the cells with Flow4AM, which basically fluoresces in the presence of intracellular calcium, and is using a different bitter molecule called denatonium. This is the most bitter molecule known to man. And so if we play this video, we're going to look at the denatonium response for intracellular calcium. Add denatonium, you see a cell respond, and then the calcium propagates to the surrounding cells. You get a little calcium oscillation. And then just to make sure that all the cells are alive, he throws on ATP to stimulate all the purinergic receptors. So to break down that experiment, he adds denatonium, a fairly delayed response, but about 8 seconds later, you see a single cell respond. That calcium then propagates to the surrounding epithelial cells, and after that minute, comes back to that initial cell. In contrast, when he throws on a purinergic agonist, all the cells light up. We did this experiment, so this is in a culture. We also did this experiment in a piece of tissue right out of the operating room. So this is a piece of mucosa from the inferior turbinate that we filleted open, and Rob was able to load up the epithelial sheet with Flow4, add denatonium. And so what we're looking at here is about a square millimeter of respiratory epithelium. And you can see that after we add denatonium, there are about 3 different locations that initially light up with that calcium. Over the ensuing minute, the calcium wave propagates until it overlaps, and then it comes back down. And just to make sure that all the cells are alive, we throw on ATP and we get a nice purinergic response with intracellular calcium throughout this epithelial sheet. So we were very interested in what are these initial cells that are responding to denatonium with this intracellular calcium. And so here's the experiment again, where we throw the denatonium on and we see this calcium propagation. But if we block gap junctions with carbonoxylone, Rob can lock that initial calcium response in that initial responding cell. And so he has about a minute, and in that ensuing minute, he came down with a sharp electrode and impaled that denatonium responsive cell. Electroporated in biocyte and lucifer yellow, so he basically tattoos the cell. And then we fix the cultures and we counterstain. And so here we're counterstaining for type 4 beta tubulin, which is a marker for motile cilia. And so in magenta, you're seeing all the ciliated cells. And here's our lucifer yellow cell. So what you can see is that it's a non-ciliated cell that has a unipolar projection. And this was very similar to the cells that were being studied in Tom Finger's lab, called a solitary chemosensory cell. So here's a solitary chemosensory cell, immunostained for that G protein gusducin in the airway, and in green are all the motile cilia. So what you see is a non-ciliated cell with a unipolar projection sticking out. So what are these solitary chemosensory cells? So in the gut, they're also referred to as tuft cells, and in the conducting airways, in the lower airways, as brush cells. And these are rare, non-ciliated epithelial cells. About 1 in 200 to 1 in 300 cells are solitary chemosensory cells. And biochemically, they look like they belong in your taste buds. They express bitter receptors, and interestingly, they co-express the sweet receptor. They express the G protein gusducin for taste transduction, and all the other canonical signaling for taste, which is phospholipase C beta 2, and that non-selective cation channel that Kevin was talking about, trypin-5. And then work again out of Tom Finger's lab has demonstrated that in the mouse nose, these solitary chemosensory cells are intimately innervated by free nerve fibers of the trigeminal nerve, and that through a cholinergic synapse, probably going through CALM1, which is exactly what Kevin was talking about, when these cells are activated by bitter molecules in the mouse nose, they induce localized neurogenic inflammation through substance P. So you get plasma extravasation and mast cell degranulation. Now I want to focus on this co-expression of the sweet receptor. And so if we do the experiment where we basically activate with denatonium, and on the y-axis, we're looking for the intracellular calcium, and we do it with 0 glucose in the bathing solution, we get a nice calcium response. As Rob increases the concentration of glucose in the extracellular space, so in the bathing solution, you start inhibiting that response. So here's 0.5 millimolar gives us about a 50% inhibition, 1 millimolar, about an 80–85% inhibition, and at 5 millimolar glucose in the bathing solution, you get nearly a 90% inhibition of this intracellular calcium response. Now, normal physiologic glucose in your mucus is about 0.5 millimolar. So right on the steepest part of this response, a little bit less, and you exaggerate this calcium response a little bit more, and you blunt it. And so we were very interested in what is this calcium response being generated by these solitary chemosensory cells doing in the respiratory epithelium. And so it did not stimulate nitric oxide like T2r38, and it did not increase ciliary beat frequency. But based on the work that we were doing with T2r38, we made the assumption that it was going to have some antimicrobial activity. And so we took our cultures and we stimulated them on the apical side with either PBS or this bitter molecule denatonium for 30 minutes. And after the 30 minutes, we basically collected the mucus and we mixed it with bacteria 1-to-1 for 2 hours. Or we took the PBS and denatonium neat, mixed it with the bacteria, let it go for 2 hours, and then plated it out in different dilutions to get CFUs. And this is the result of that experiment. Here's the PBS treated mucus where we get a nice bacterial lawn. As we dilute it out, we can actually count colonies. But in the denatonium treated cultures, the ensuing airway surface liquid, there was no viable bacteria. And I will tell you that when you just take denatonium straight up and you mix it with the bacteria, it doesn't have any bactericidal effects. So we have this very robust mucus after it's been stimulated with denatonium. If we look at it and see what's expressed there, so here's Page SDS, just a simple protein gel, 24 hours of secretions from the top of those cultures. We then wash them off and stimulate for 30 minutes with PBS or 30 minutes with denatonium. We see a whopping band down at the bottom of the gel between 4 and 6 kilodaltons. And based on this and the prior result, we make the assumption that these are antimicrobial peptides. And so here's an ELISA looking at the mucus after we stimulate with PBS. When we throw on denatonium, you get a 300-fold increase in the amount of defensins in the mucus. If we block the gap junction, so we don't let the calcium propagate to the surrounding cells, we blunt that. If we do it in the presence of glucose, we blunt it too. Now, interestingly, even if we do it in the presence of a sweet receptor agonist, sucralose, but we throw in their lactazole, which is a sweet receptor antagonist, we can reverse the effects of the glucose inhibiting the calcium response. And here's a little bit more pharmacology. If we block phospholipase beta 2, we inhibit it. If we do it with 0 calcium in the bathing solution, we also inhibit it. We found other bitter molecules that do the same thing. So here's absinthin, parthenolide, amorogentin, all induce antimicrobial peptide secretion from the sinonasal epithelium. Interestingly, phenolphthaleocarbamide and C4HSL, these are those agonists for T2r38. So the ciliated bitter taste receptors, when you stimulate them, they do not release antimicrobial peptides. So we generate the following model, that peppered throughout the respiratory epithelium are these non-ciliated solitary chemosensory cells every 100 to 150 microns that co-express bitter receptors and sweet receptors. When these bitter receptors are stimulated by, we assume, a microbial compound, you get an increase in intracellular calcium, it spreads through the gap junctions and induces the surrounding epithelial cells to release antimicrobial peptides. However, that whole response is governed by the sweet receptor. And as long as there's physiologic glucose bathing these cells in the mucus, the sweet receptor is stimulated and is inhibiting the propagation of the bitter receptor signaling. And what we're proposing is that as the colonizing bacteria proliferate, they're going to consume the glucose. As the glucose levels drop, this break is relieved, and some of the calcium is spread out, and you release some antimicrobial peptides. And so we think that the solitary chemosensory cells are literally tasting the glucose in the bathing mucus as an indirect measurement for bacterial density. And as the bacteria proliferate and the density goes up, the glucose goes down, you release some antimicrobial peptides to do localized population control of the bacteria. Now, clinically, we started looking at the amount of glucose in the patients that we were operating on. And so here's glucose in all of the patients that don't have any infection or inflammation in their nose. And I neglected to mention, we do a lot of combined cases with neurosurgery where we use the nose as an access to the pituitary or other skull-based lesions. And so we get a lot of tissue, and we're operating on a lot of patients that don't necessarily have chronic sinusitis. So here, our control patients are sitting right at 0.5 millimolar. And our sinusitis patients, whether they have type 2 inflammation or type 1 inflammation, are sitting at about 4 to 5 times the amount of glucose that's sitting in their mucus. And that's most likely because of the persistent inflammation creating a leaky epithelial barrier, and glucose is leaking out. And I'll just put this dose response back up here. So that our controls are sitting right here on the yellow curve. A little bit more glucose and they inhibit, a little bit less glucose and they exaggerate. But our chronic sinusitis patients are tonically living down here. And so they're tonically repressing or inhibiting the ability of the bitter receptors to do what they're doing. Now, I want to finish up with a little bit more of a translational component, and going back to that first question with regard to, can we potentially leverage what I just told you about on these taste receptors in the sinonasal epithelium as a therapy? And so one option would be to activate the bitter receptors on the ciliated cells to drive local NO, which, as I showed you, has an increase in mucociliary clearance and has direct bacterial killing activity. Alternatively, we could focus on trying to stimulate the bitter receptors on the solitary chemosensory cells to drive antimicrobial peptides. We can do that either alone, or we can do it in conjunction with lactazole to block that sweet receptor. And remember that our sinusitis patients have a lot more glucose up here. Alternatively, and especially in lieu of the fact that these cells also generate neurogenic inflammation when you activate the bitter receptor, maybe just using lactazole alone to block that excess glucose may then bring these cells back to normal physiologic function. So two therapeutic options to explore. And we've really focused on the nitric oxide pathway. As a proof of principle, we took some individuals that were genotyped for T2r38, so a homozygote functional, non-functional heterozygote, and we squirted that bitter molecule PTC in their nose. And here on the y-axis, we're looking at exhaled nasal NO. Now, this is different than when you think about exhaled NO from the lungs. In this experiment, patients actually hold their breath, and you're aspirating at a slow rate the amount of air out of the nose. And so you establish a baseline, and then you usually run the experiment for about 30 seconds, because again, the patient is holding their breath. So here at baseline, it's about 500 parts per billion. You squirt PTC into the nose, and within 3 minutes, you get a near doubling in a homozygote functional, as expected, of exhaled nasal NO. In a heterozygote individual, there's a little bit bump, and in a homozygote non-functional, you see absolutely no increase in exhaled nasal NO after we put PTC in. So it's a good proof of principle idea, but from a therapy, it's limited because it's only really going to be useful in homozygote functional individuals, and those patients really probably don't even need this therapy. So what about focusing on other bitter taste receptors? Well, we could stimulate T2r4 or T2r16, but we're going to run into the same problems, that they have these common functional or common non-functional polymorphisms. So luckily, there are over 550 compounds that have been identified as having a bitter taste. There are probably many more that have yet to be identified. And we have several different options in driving a therapeutic program here. One could be individualized therapy. So we have a patient taste 6 different bitter molecules that activate bitter taste receptors on the ciliated cells. And whatever they taste as most bitter, well, that's going to have probably the most profound effect in driving the nitric oxide. So we could basically individualize the therapy. It's very in vogue these days. Another option would be to make a mixture of bitters so that you're hitting multiple bitter taste receptors and thereby minimizing the genetic effect. From a regulatory pathway, it's a little bit more difficult to work with mixtures. Or the other option is we could find a molecule that's promiscuous and activates multiple bitter taste receptors. And we kind of stumbled on this because we were looking for a placebo for PTC, and we tested quinine. And the problem with quinine is that we got incredibly robust epithelial NO generation. Here you can see a dose response to NO, and here's ciliary beat frequency, where we get about a 10% increase in ciliary beat frequency. And I should also say that it's not a direct relationship between ciliary beat frequency and mucociliary clearance. It's closer to an exponential. So a 10% increase in ciliary beat frequency has a robust effect on clearing mucus out of your nose. And the other reason why we like quinine is it's promiscuous, activates up to 9 bitter taste receptors. And then the last thing about quinine is it's already been used in the human airway in multiple trials as, ironically, a placebo for several antibiotics. And this is what happens when you put quinine in the human nose. So here's 30 runs of baseline at about 500 parts per billion. Put quinine in the nose, and within 3 minutes, we get a near doubling in exhaled nasal NO. So based on this data and a little bit more data, in conjunction with Gene 1 Life Sciences, so we partnered through PCI with Gene 1, We filed an IND and got approval, and we were about to go into a quinine topical nasal spray to prevent post-op sinusitis. And as we were getting ready to start enrollment, COVID happened and our trial got placed on hold. And so like everyone else, we started reading a lot about coronavirus and came across a bunch of literature from 2005, 2006, based on the first SARS, demonstrating that NO has pretty robust in vitro effects against coronavirus. And so we did a little bit more preclinical work and went back to the FDA, since we already had all this product at the Investigational Drug Service, and we repurposed our trial for sinusitis as a prophylactic treatment for SARS-CoV-2. And that's ongoing right now, and we don't have results yet. But hopefully in a couple months, we'll know if there's any efficacy to squirting a bitter taste agonist up your nose to try to prevent coronavirus infection. So in summary, what have I told you so far? Taste receptors are expressed at multiple mucosal barriers. Endogenous ligands for these T2Rs appear to be microbial products. That there's an emerging role for these taste receptors in innate defense that's complementary, and I would probably say based on the time of response upstream of the toll-like receptors. And from a clinical perspective, what really fascinates me about this is that there are these common functional polymorphisms or non-functional polymorphisms that may explain why certain individuals are more likely to get recurrent infections with a specific organism. And expanded out may actually help define an individual's microbiome. We also think that taste testing may prove clinical utility and be quicker than genotyping our patients. And as I showed you with the last couple slides, we think that topical bitters may have therapeutic implications, at least in the upper airway, but could be expanded to other mucosal surfaces. And so in summary, I just want to thank all of my collaborators. So this is Penn Rhinology. I showed you most of the work that Rob Lee was doing. Mike Kohansky is my junior partner now, both in the lab and clinically. The surgeons that are constantly generating all the tissue for us, my collaborators over at the vet school, all of my collaborators over at Monell, and our partners in industry, Joel Maslow, Christine Roberts at Gene One Life Sciences. And with that, I will conclude and take any questions. Thank you.
Thank you so much, Noam. I really appreciated that. We have about 3 or 4 minutes for live questions, and I'll take the first question as chair's prerogative. So there are many microbial products. And so what is your sense of all of the bitter receptors? How much do we know and how much don't we know about which microbial products are ligands for these receptors?
So we know that in the urinary system, the solitary chemosensory cells respond to a product made by E. coli. I am unaware of which bitter taste receptor is doing it, but there's another model with another organism in a completely different mucosal surface. We have been running a large project looking at the microbiome in the context of all of the polymorphisms of the taste receptors. And what we're trying to identify is whether there are specific non-functional polymorphisms that associate with a specific organism in the nose. We have a couple hits. It's not really powered large enough, but it looks like T2r60 may correlate with Staph aureus infection in the nose. Yeah, so I'm not sure where else to go with that, Danny. But yeah, there are other microbial ligands that have been shown to activate these bitter taste receptors.
So you're welcome now to unmute yourself and ask a live question if you're so moved. You can also, in the reactions panel at the bottom, you can raise your blue hand and I'll call on you. Or your other alternative is to do what I see you're all doing is questions are appearing, popping up in the chat section for Noam, and he will be addressing those directly. I see that Michael Beers has his hand up. Michael, please ask your question.
No, great talk. Quick question in terms of repurposing quinine. Does it work against other viruses in the coronavirus family? So could you use this as a prophylaxis for the common cold or before we all go on an airplane, you know, for how clean they are these days?
So that's one thing we're going to be looking at with Susan Weiss. She's got a whole bunch of viruses that we're going to now, we have an in vitro platform that we can test this on. So we haven't gotten there yet. Literally the funding just came through yesterday with the MOU. So that is one of the things that we're going to be talking about with Susan. She wants to focus on all the coronaviruses. I'm specifically interested in rhinovirus and influenza.
Okay, Noam, thank you for that. We are now nearly at the top of the hour, so that leaves me to thank Noam Cohen very much for a very stimulating keynote talk. And my next duty with my introduce a Monell scientist, Marco Tizzano, and he'll be talking about gingival cells and the role of T2Rs in that immunity in that cell type. So Marco, please go ahead. We can see your slides.
Okay. Let's see if I can find the pointer here. Thanks for having me here. It's an honor to be able to show again my exciting research on solitary chemosensory cells that were well introduced by Noam Cohen and now from Peihua, and in general for the pathway of taste that has been described previous from our colleagues. So my talk today will be centered on a new location for these chemosensory cells that have been found in the gingiva. And again, as explained from my colleagues, these are involved in as immune sentinel for bacteria, in this case in the mouth and the periodontal disease. So you can see what was introduced before about the taste system. It's located in the tongue. There is taste cells all over the places, and we talk about these extensively during this symposium. And what I want you the attention on for this particular case is that chemosensory cells are located all over the body, as you can see in this schematic, but they are also found with taste receptor in SCCs or not, in both nasal, main olfactory epithelial, kidney, testes, and the list keep going on, and including gingiva, as I will show you in a second. But what I want you to remember about this is the pathway of these taste cells in extraoral locations. So they have taste receptor, they follow gasducin, as you can see also here. Then there is activation of PLC beta 2, calcium, increase, TRPM5 channel, and then a release of possible neurotransmitter or modulator, as my colleagues also pointed out. So remembering this, most of my work is based on knockout animals that block either the gasducin pathway, the TRPM5 pathway, or the absence of SCCs with the skin 1A knockout that also Peihua mentioned. So in general, what we can say about SCCs, and just to summarize that and giving a final touch to that, is that accumulative evidence that you can see many here, but there is even more, suggests that activation of T2r mediated signaling in these SCCs or similar chemosensory cells in this location of the body that we mentioned a second ago has a major contribution to local mucosas innate immunity. In particular, when SCCs in the body are activated by compound produced by bacteria or pathogens in general, like also, parasites, they seem to have a common function. They detect bacteria or this parasite and evoke inflammatory and immune response to remove the pathogen. So this is in general the concept, and I want to show you how this is applicable to the gingiva. So here is a schematic of the mouse head. You have respiratory epithelium, olfactory epithelium, two locations where SCCs are of two different types, microvilli cell here and solitary chemosensory cell in the respiratory. On the tongue, you have taste cells, but then on this portion of the mouth, where the gingiva around this molar is, we found, again, GFP expressing cells that are TRPM5 positive. And if you, take one of these group of cells and slice in the gingiva there, you will find a group of gasducin or PSC beta 2 positive cells that are also expressing TRPM5. So they have all the signaling cascade that are actually present on the surface facing the tooth. The T is the tooth part, and this part is the outside of the gingiva. And you can see how this area is highly, have an highly presence of these SCCs. And actually there is where the pocket of periodontal bacteria will be present when you have a problem with that disease. When you take out this part of the gingiva, you will have presence of, again, gasducin triple 5 PSC beta that are signaling cascade, elements of the taste transduction cascade, and then you have all these taste receptors. Not all the taste receptors are present in gingiva, but a good part of them. And when we take these taste receptors and test them in hex cells, you can see how the bacterial colon sensing molecule and several bitter compounds, for example, including the natonium here, cyclohexamide, and then the lazide that is a bacterial compound, or the artificial one that are acetylmonosylilactone C6 or C8 or C12. So you can see how testing all the taste receptor that we found present there, in particular one, the T2r 105, is highly responsive to the bacterial compounds, but also to the natonium and cyclohexamide. They do that in a dose response curve for the bacterial compound. And when we take isolated cell from the gingiva that express 3.5 GFP here in the bottom panel to do experiment of live calcium imaging on isolated cells, you can see how these green fluorescent GFP 3.5 isolated cells respond to the natonium, to C12, again the natonium, and so on to different concentration of C12. They also respond to the cyclohexamide, and they do not respond to, for example, the diluent of these bacterial compounds that are pretty much hydrophobic, so you need to use a diluent for them. Going on, what we found is we use a gasducin knockout mouse to show if there was any evidence of a difference in a periodontal status in the gingiva, that is the place where we think these cells are more pertinent for this disease. You can see how GNAT3 mice that are the gasducin knockout mice have a natural occurring alveolar bone loss that is accelerated. What that means, so if you check a wild type at 8 weeks of age and then at 16 weeks of age, you take their molar area with the bone surrounding them, you see how this area in yellow here, it's increasing with the age. That is natural in most of the people. So with the age, you will see more of your bone exposing the tooth, and you will have what is starting to call a pre-periodontal problem. And then in the gasducin knockout, you see that the similar starting 8 weeks of age, that is kind of the young age we can consider in mice. And then you can see as 16 weeks as the age progresses, there is an increase of that you can quantify here, that there is a significantly lower alveolar bone loss, but still not as bad as you can expect with later in the age. This difference is not visible in a micro CT scan, meaning that there is not a so big difference at this stage in terms of bone loss, but you start to see a bone level difference. And this bone level difference is also comparable to the oral microbiota composition that also change in these knockout mice. So if you see that the absence of gasducin is a marked effect on the oral microbiota composition, and you can see how already an important difference on the oral bacterial composition is visible and occur although you don't have any alveolar bone loss. This was promising, so we thought, can we accelerate this process? So we applied a ligature-induced periodontitis model. What this means in very simple word is that you apply a ligature around one tooth, similar to when you have some food stuck in between your molars and it's starting to give you some pain and inflammation. We cause this for a week in a mice, meaning that bacteria accumulate around this ligature, cause some inflammation, and you accelerate the process of periodontal disease. So it's like an aggressive periodontitis. And when you do this ligature around the tooth of a wild type, you see that there is an increase in alveolar bone level that, if you remember from before, it was very small starting even at 16 weeks. So if you check this difference here, quantify here in a bunch of wild type mice, that increased drastically in gasducin knockout, 3.5 knockout, and skin 1A that are missing SCCs. You can see how the bone is more exposing more the root of the teeth, that is the measure of this alveolar bone level that eventually can cause bone loss, as you can see in some of these. this difference go along with the amount of bacteria. So when we measure the amount of bacteria around that ligature, you can see how the bacteria amount is also increasing. So there is more bacteria and more bone loss. We also quantified that with the micro-CT, and you can see here a typical wild-type micro-CT versus a gas-induced knockout micro-CT. You see more bone loss that we also analyze with an automatic method that now Hong Wang, my lab, is developing. So you can see as in a ligature side, the green area is still bone and is still present. When you check a ligature wild-type or knockout, you see a disappearance of that green part that is now in yellow here. And in the knockout mouse is even more. So when we quantify that, there is a bone volume, but particular bone density that is lost in these mice. What is the cause of that? Practically, there is more inflammation in the knockouts. You can see here, there is more marker of inflammation. In the same time, there is something that also Noam mentioned, that there is antimicrobial production by SCCs. And if you see in the wild type, there is a normal amount of production of this defensin, but the knockout mouse is not able to produce many of them. And so in conjunction with this, we can say that there is more inflammation, less antimicrobial that can be considered together and justify the increase of bacteria in the mouth. The composition of the microbiome, as we say in this ligature, it's clearly separating gas-induced knockout from the wild types, both in terms of beta diversity and so also microbial network. The alpha diversity is also different and is lower in the ligature from the gas-induced knockout. And when you check exactly the composition of this bacteria in the ligature, you can see that they are different in the knockout and wild type. There is an increase in particular of Pasteurella in gas-induced knockouts, and the major component of that increase is this NI 1060. That is a gram-negative bacteria that is a specific bacterium correlated to the human AA bacteria that is actually the pathogen for an aggressive form of periodontitis. As we said before, the ligature causes that. So it's very similar to the type of bacteria that is also present in human. Then we decided to go over, as Noam Cohen mentioned, we wanted to use a ligand for these SCCs as a possible hack for activating artificially these SCCs. So we use a T2r 105 ligand that is denatonium, that we were able to boost the expression of antimicrobial peptides. So as you can see, remember here, there is a response to them in this taste receptor. We took mice, wild type and knockout, exposed to PBS and denatonium in 2 groups. For 7 days, we topically applied PBS or denatonium twice a day, and then we euthanized the mice. And what you can see in general is that the defensin B3, it's very increased in the wild type with denatonium, but stayed low in the gas-induced knockout, meaning that it cannot detect that because it's missing the gas-inducing portion of the transmission signal. So then we decided to do a ligature in the same identical setup of the experiment to accelerate the, the periodontitis. And what you can see is that similar as before, wild type we have some bone level loss, and the gas-induced knockout we have that increased. And you can see here in the graph both the increase in bone level loss and also the bacterial amount. But then when you check in the, no surprisingly, the gas-induced knockout, you don't have an effect because denatonium is not able to stimulate this type of animal because it's missing the gas-inducing. And you see that both alveolar bone loss and bacteria stay the same. But then surprisingly, when you take that in consideration in denatonium in wild types, you see that the alveolar bone level is reduced as a loss, but also the bacterial amount is reduced. So this is a very potential component that we want to consider for therapeutic and also preventive medicine for periodontitis. So in general, in the summary, SCCs in the gingiva are centered for bacterial invasion. They use the T2r mediated signaling cascade to detect this bacteria. The activation of this cascade of signal as a major contribution to the gingival innate immunity, stimulating the release of antimicrobial peptides that will control these bacteria. And then the absence of some molecule in the GSCCs or the absence of GSCC in the skin 1A knockout can increase alveolar bone loss, alter the oral microbiome as we showed you, but also cause a more severe periodontitis. And clearly, stimulation of this pathway in the SCCs can be hacked to promote gingival epithelial health and to activate this cell to produce more antimicrobial peptide and consequently reduce the oral bacterial growth. I want to thank everybody in the Tizzano lab and the Margoski lab, because this was a job done in collaboration with Dr. Margoski, in particular Shin, Jiansu, and Rangwi that has been running most of the experiment. And thanks for my funding opportunity.
Marco, thank you so much for the talk. We really appreciate that. I would direct all the people that want to ask questions to the chat, so Marco will be available to answer your questions. I see a tough time ahead for people making a mouthwash and toothpaste with all those bitter ligands necessary. So I turn now to introduce Dr. Bruce Kimball. He's a scientist at the Monell Chemical Senses Center, and he's going to talk to us about how airborne volatiles from animals can be used in illness detection. So Bruce, go ahead.
Thank you, Danny. I appreciate being invited. I hope when I'm done that you appreciate that you invited me. If it isn't going to be evident at the end, I know nothing of immunology, so I'm going to throw that out there right now. My area is working in the analysis and interpretation of the volatile metabolome. It seems in science, particularly in biology, that you have to work in an ohm to be taken seriously, and so I believe the small molecule people were very jealous of the genomic people and the proteomic people, so they decided to label all the metabolic products and intermediates as the metabolome. And you can just imagine there's a lot of them. Here's a schematic of just pyruvate metabolism, and just spread throughout here are compounds that you could measure in all kinds of body tissues. There are probably greater than 40,000 of these things, and it's because there's so many of these metabolic pathways, some that feed into others and output to others. And then you also have all the exogenous things that might be part of a diet or exposure to things. So there are a lot of metabolites in this system. Of the greater than 40,000, less than 2,000 of them are actually volatile, what we would consider volatile, that you could measure in the headspace or present in the atmosphere and are available to be detected by olfaction. And they can be found in all sorts of compartments. I tend to work a lot in urine. It's a nice area that's considered to be sort of a window into metabolic pathways. Some people with, I guess, with omic envy have labeled this the Volatile. I don't consider that to be, I think that's a little ridiculous, so I'm just going to talk about the volatile metabolome. So I think it's no mystery, or people realize that metabolism is impacted by health. It's all sorts of systems are disrupted by infection, injury, malignancy, even immunization we have found. And it makes sense because many of these products of metabolism are the currency which allows the other processes to occur. Mounting a detoxification or an immune response is costly. That has to come from somewhere. And so you can just imagine with all of these different metabolic processes that there are going to be changes in outcomes. And the thing we also have to remember is the products of our metabolism aren't just affecting us. They're affecting all the microbiota that we harbor. And so you almost have to think in terms of there being this ecosystem that when a certain metabolite becomes more prevalent, some organism in the host might be able to take advantage of that. And we in fact found that with pyruvate and avian influenza in waterfowl, that when glycolysis was upset and more pyruvate was being produced by infected ducks, certain microorganisms in their gut were reacting to this abundance of pyruvate that wasn't there, and they were able to thrive, and their metabolism actually dictated what we were seeing in duck feces. So I kind of want to throw out a little, maybe a caveat about the value of looking at the volatile metabolites in response to some sort of insult or injury, and that is alteration of just a single volatile has been incredibly rare. We've never actually seen that only one metabolite is altered by an event. And probably more unfortunate is that production of a unique volatile is incredibly rare. It would be wonderful if every disease, infection, injury had its own signature volatile that would only appear in the case of the event of that occurrence. Well, that just doesn't happen. So the consequences of these means that health conditions are typically predicted by patterns of volatiles, and we have to look at how patterns of volatiles are altered. And it's up and down regulation of those metabolites that are present in the healthy state. So you have to know something a little bit about what the unaltered condition is. How do we observe these alterations? It's just like olfaction, right? We have to detect them, and we use a number of instrumental tools to detect these many volatiles. And then you need to learn about them. So, we use in the chemometric approach a number of statistical approaches to analyze those patterns. I'm not going to get into the weeds on chemical analysis, but suffice it to say, we use dynamic headspace gas chromatography with mass spectrometry to generate these chromatograms. And so we can work with model systems, we can work with the actual target organism, be that human or wildlife, and collect samples and do a lot of chemical analysis. And so you might imagine we end up with a lot of chromatograms, but one chromatographic trace has a heck of a lot of data in it. Just one chromatogram probably has over 2 million data points, because the mass spectrometer is taking a scan at many points along this chromatogram, and each scan has many hundreds of data points for all the masses that it's monitoring. So we need to get rid of the noise. We need to align all the peaks over time. We don't tend to do all this work in 1 week, and so over many months, some of these chromatograms, the peaks will shift a little bit, so we have to line those all up. And then we have to come up with a semi-quantitative value for what that peak represents. So we need to integrate the area under that peak or calculate its height and then standardize it against something. In this case, we put an internal standard in every sample. And then we use a variety of pattern recognition techniques, machine learning, random forests. I use a lot of LDA just because of my rudimentary approach to statistics, but it actually ends up being pretty powerful. So I mentioned that we use instrumental techniques and statistics for detection and learning, but obviously the mammalian system, well, all living systems have figured out a way to detect things and recognize what that is. And so we can actually use a nose and the brain to do this. And so I have an example here, a little video. These 5 boxes contain duck feces, and I'll let you know that this far one on the right is the one that contains feces from a duck that was experimentally infected with avian influenza, and the other 4 are from healthy ducks. And we're going to let our nose run in here and tell us which one is the infected source. And so the scratch at the box tells us he chose box number 5. And I like to show this one because Sometimes the four legs get involved along with the nose. And this was during the learning of the task. And this one got very excited trying to figure out which one he was supposed to find. So if you look in my lab, and this includes my laboratory at USDA in Fort Collins, Colorado, we've done a lot of work looking at model systems and looking at actual wildlife concerns. So we've studied using trained biosensors, and that includes ferret, as you just saw, but mice in a Y maze and dogs in our lab in Fort Collins. Along with the chemometric approach, we've looked at immunization and infection and injury and genetic precursors to conditions like Alzheimer's in a number of species. I do want to mention that our late colleague George Preddy also had initiated a really wonderful collaboration with Cindy Otto and Charlie Johnson at Penn looking at human cancers. And Cindy Otto had her working dog group looking at this, and Charlie Johnson has his really nice sensor systems working at this. And George was doing the chemometric part in the lab, and I'm doing my best to continue that work. So I wanted to acknowledge that that's happening at Monell too. So what about immunology? I admitted I'm not an immunologist, and presumably many of you would like to hear something about immunology and the volatile metabolome. We did try to kind of get at what is the mechanism for these alterations. Certainly the immune system has to have a role here. And so we started looking at kind of the initiation of the whole immune cascade and using the mouse model with in vivo presentation of pathogen associated molecules that represent different classes of bacteria and viruses, we're able to promote a cellular immune response, collect the urine, and do the chemometric analysis and determine if there is a difference based at least on these broad classifications of pathogens. And so, forgive my very rudimentary cell here with a toll-like receptor, but when a detection of this case with TLR4 of a gram-negative bacterium, this cascade of events happens, right? There's cytokine and chemokine production, a whole inflammatory response, but there's also an alteration in the volatiles that we can measure in urine. And then we can just repeat this with the other pathogen type things. Here, representative of the gram-positive bacteria, and then also representative of RNA being introduced into the cell by a virus. And we can then compare using animals or chemometric analyses. How do these volatile patterns differ, and can we discriminate amongst those? And in fact, we can. So we're actually embarking now directly in this area here to look at can we inform clinicians about the source of an infection to perhaps facilitate better use of antibiotics. And that's a really nice new thing we're starting with my colleagues at Children's Hospital that's funded by the Kleberg Foundation. So where else are we going with this? Advances in chemical analyses and sensors are happening all the time. We can always do better, and we have a guidepost. We have that animal nose. We want to be as good as the mouse or the ferret or the dog. And so those types of instrumental advances are very important, and we need to keep up on that. But I think one thing that gets lost in all this, that it's just not the olfactory sensory neurons that are doing work in those animals and in us. There is a brain involved, and we need to be more cognizant of how pattern recognition is happening in these animals. And so this area of neuromorphic computing, I think, is a very exciting area in which we might be able to do a much better job to discriminate amongst the patterns that we observe. The other question is, I kind of showed you in the cellular response that, that we can tell the difference between some broad classes of pathogens. But, immunology being what it is, and particularly with an adaptive response, there is this, the hope that there might be some real pathogen specificity. Right now, well, for quite some time now, there's been a lot of interest in what the possible odor of SARS-CoV-2 infection might be. The fact that infection causes an alteration of the odors emanated by an infected person is really not surprising. But the greater question is, how does that differ from infection with, say, influenza A or human coronavirus? How much specificity is there? That is like a really important question that I think we need to have our eye on as we move forward. And then the other thing is, what kind of devices are we going to use? I'm not opposed to having a ferret diagnose my health, but I think a lot of people might find that a little disturbing. So we need clinical tools, and where we would like to do this, I think most simply would be wonderful, like a dipstick thing that we could use in urine or saliva that could tell us something about the source of the infection. But I think electronic noses are also they're going to be more expensive and maybe not as available in low resource settings. But I think that's an area also in which we can take the ferret without using the animal. Excuse me. So I do want to acknowledge my colleague Gary Beecham and members, current and former members of my lab, and quite a bit of funding from USDA and some other funding from the state and DoD for traumatic brain injury work, and then the Kleberg Foundation, which is funding our new initiative. So with that, I thank you for your attention.
Bruce Kimball, thank you very much for that talk. I would ask you to direct your questions to the chat in the interests of time, and I now turn the chair duties over to Kevin Foskett, who will introduce the next speaker.
Yeah, thank you, Danny. The next speaker is Dr. Andrew Vaughan, who's at Penn. Andrew, if you're set, go ahead and take it away.
Sure am, and I'm just in the process of sharing my screen. You guys can hear me okay?
Yes. Mm-hmm.
Okay, great. And you can see my screen?
Yes, we can.
All right. Wow. Amazing. Thanks so much for the invite to this awesome forum today. As program notes, I'm going to tell you guys about our unexpected identification of tough cells arising in the distal lung, where really there are no tough cells, at least in naive uninjured lungs, in response to influenza infection, and what we think this might mean for chronic lung pathology. So really, my lab here at Penn is about 4 years old, and we largely focus on lung repair and regeneration. We've looked a little bit at the endothelium more recently, but largely we focus on the epithelium. And in order to study repair of the epithelium, we use H1N1 influenza virus, mouse adapted. We like this particular model because we can see multiple modes of lung regeneration happening in the tissue concurrently. So laser pointer. Here you can see in this section of lung, these cells in red. These are alveolar type 2 cells. And really since the 1970s, these cells have been known to act as a facultative stem cell. They can proliferate, they can differentiate into gas exchanging type 1 cells. So these cells really act as a facultative stem cell and are pretty good at regenerating the lung after mild to moderate to mild lung injury. However, there are other regions of the lung that aren't so lucky, where these type 2 cells have essentially been ablated. And in these regions, we see these cells in green. What we're actually standing for in green here is cytokeratin 5. This is a marker of basal cells in a number of different organs and tissues. In this case, the basal cells, at least in the mouse lung, are actually restricted to the trachea. So these cells are ectopic to the alveolar epithelium. And starting as a postdoc and continuing on into my current lab, we've really shown these cytokeratin 5 expressing cells are very poor at ever differentiating into normal lung alveolar cell types. So they sort of serve emergency barrier restoration function, but are really bad at ever giving rise to the types of cells you really need for normal lung function. And it's permanent. We see a year after infection, a persistent loss of pulmonary function, and this plastic regions of the lung really never resolve. I just wanted to very last minute bring in that we recently have demonstrated that this does in fact happen as a result of COVID-induced ARDS and diffuse alveolar damage as well, where we're looking at the lung from a patient who was on ECMO over 2 months with really severe COVID-19. And we see this sort of epithelial dysplasia marked by these kind of 5 cells in really large fractions of what should have been alveolar. Just a little higher magnification. So really, this has remained a focus of my lab since we think the presence of this epithelial dysplasia is contributing to long-term dysfunction. And over the course of studying this, we realized that these basal cell-like dysplasia was not persisting in a vacuum. That we have these cytokeratin 5 cells present at least a year post-infection, but concurrently with this, there's also increased numbers of inflammatory cells, just marked CD45 here, which is pan all immune cells. And this is really strange because the virus itself is cleared by about a week and a half post-infection, so we don't know what's driving this. But we thought, well, perhaps the epithelial dysplasia is driving the chronic inflammation, or vice versa. Maybe these aren't unrelated. So, despite the fact that I'm very much not an immunologist, we decided we better characterize this a little bit more carefully. And much to our surprise, at least at later time points after flu infection, so about 3 weeks on, we see markers of type 2 inflammation, IL-13, IL-25. Weirdly, we see eosinophils that are taking up residence in the tissue, as well as goblet cell metaplasia, another hallmark of type 2 inflammation, both within the airways as well as these remodeled regions of the lung, this epithelial dysplasia, which is really airway-like cells present in the alveoli. Just looking for some markers of goblet cells. So with this in mind, we went out on kind of a wild hair and asked whether tough cells might be involved in this. And luckily, due to some great work that was just showed, I don't have to give you guys too much of an explanation on tough cells. But I'll just briefly review that they've really been, their function has really been best characterized now in the small intestine, where they've been shown to be the sole producers of IL-25. And utilizing these gustatory signaling pathways, they can respond to various types of infection, where they release IL-25. This initiates a feed-forward loop, where the IL-25 acts on ILC2s, ILC2s produce IL-13, IL-13 then acts on the stem cells of the gut and promotes increased differentiation of tough cells. So it's a feed-forward loop, more tough cells. Also more goblet cells, and indeed hyperplasia of the entire small intestine, where the small intestine gets 25% longer within a week. So with this in mind, we asked, okay, is there a role for tough cells in this chronic inflammation or lung remodeling? And I should say at baseline, there are no tough cells in the lung proper. There are tough cells in the trachea. They're also called brush cells up there, or solitary chemosensory cells. In my opinion, I think these are really all the same cell type, just present in different anatomic regions. At day 0, there's none of these in the distal lung. We start to see the appearance of the basal-like cells, still no tough cells. Then by about 2 weeks post-infection, we start to see a few cells staining intensely with this canonical, or at least in mouse, tough cell marker DCLK1, a few more, and then by 3 weeks post-flu, it is quite a different story, where these dysplastic remodeled regions of the lung are really littered with these DCLK1 positive cells. We can zoom in, we can see they possess this sort of canonical tough cell morphology. I hope you would agree these are very similar in appearance to what Noam and some of the other presenters showed for a classical tough cell. Of course, I should say that these tough cells in these post-influenza lungs always seem to arise either within or right next to those dysplastic remodeled keratin 5 expressing regions of the lung. which suggested to us that they might share a cell of origin. Now, as a postdoc, I had shown that essentially all of that epithelial dysplasia is derived from rare basal-like cells that are present in the intralobular airways. And then upon infection, they expand like crazy, proliferate, migrate, and are the sole source of all of this dysplasia. This has been confirmed by Wellington Cardozo's group as well, that these rare cells give rise to all of this dysplasia. So with that in mind, we asked whether the tough cells were derived from these same progenitor cells. And in fact, by this lineage trace, they are. Really, we couldn't find any tough cells that weren't lineage traced. So they're essentially 100% derived from these intrapulmonary basal-like cells. So everything I've shown you up till now has been really based on one marker gene expression, DCLK1. So in order to really prove that these are what we think they are, we utilize this TRPM5 GFP reporter, which now everybody knows what TRPM5 does, and this reporter is really quite a robust reporter for it. There are, again, none of these TRPM5 GFP positive cells present in the lung at baseline, but post-infection, we see all these green cells popping up. And essentially all of the DCLK1 positive cells are TRPM5 GFP positive. There are a few GFP positive cells that don't express so much DCLK1, suggesting some heterogeneity here, which I'll get back to. Also, all of these TRPM5 cells that arise are P2F3 positive, the tough cell transcription factor that's really required for the adoption of the tough cell. So we can capitalize on this GFP reporter. We can flow out these tough cells from the post-flu lungs. And we've done bulk RNA-seq, which I'm not going to show you, but we've also done single-cell RNA-seq. Interestingly, we actually capture not only mature tough cells in this fashion, but also the basal-like cells that are beginning to differentiate into the tough cells. They co-express some of these markers even while they're sort of still in this hybrid basal tough cell state. And they express the gustatory signaling genes as well as leukotriene synthesis, which hasn't been discussed too much yet, but I'll talk a little bit more about that later. And again, P2F3. So one of the questions we had initially, of course, is, are we really just recapitulating the same circuit that's known to exist in the small intestine in the lung? In other words, is the expansion of these tough cells IL-13 and IL-25 dependent? So in collaboration with Maya Kotas, who is a pulmonary fellow in Rich Loxley's lab at UCSF, we infected IL-25 knockout mice as well as IL-13 receptor knockout mice, and finally P2F3 knockout mice. And in the case of both the IL-13 receptor and IL-25 itself, these were really indistinguishable from wild type. Tough cells still rose in roughly equal numbers, no obvious change there at all. However, of course, post-flu tough cells are still dependent on P2F3 as we would have predicted. So this is interesting and really suggests to us that a fundamentally different circuit is at play in the post-influenza lung than what is happening upon, say, hookworm infection in the gut or delivery of sesame. But of course, what we don't know and what we hope to find out in future directions is what the proximal signals are required for these post-influenza pulmonary tough cells to arise. So the other sort of side of the coin here is what is going to be the consequence of having these increased numbers of tough cells in the post-viral lung. And I should say, we don't know the answer to that yet. We're actively investigating that. But I think in order to think about what the effects might be, we should review what the potential inputs and outputs of these tough cells are, because I think they can give us some clues. So we discussed how they are the sole producers of IL-25, at least in mice, and can participate in type 2 immunity in that way. So perhaps they're involved in type 2 immunopathologies like asthma. Marco Tizzano and some others have really nicely shown that these cells are cholinergic. They can participate in neuroinflammation, presumably other cholinergic effects. These cells may also be involved in triggering the release of antimicrobial peptides from adjacent cells that Noam Cohen has nicely elucidated. And finally, there's recently been some really fantastic work from Nora Barrett's group showing that these tough cells are enriched in all of the enzymes necessary to produce eicosanoids, and in particular leukotrienes, which again are really intimately involved in type 2 immunity and asthma in the lung. So with that, we really are at the point of thinking about what these cells do in the post-flu lung. We are still, of course, very interested in what signals are required for the development of these tough cells in the first place, since they appear to be independent of IL-13 and IL-25 feed-forward loop. And then in terms of the function, we're interested in whether the presence of tough cells might impact the persistence of this epithelial dysplasia. Since in the gut, activation of tough cells promotes proliferation of the surrounding or adjacent epithelium. Perhaps that's why this keratin 5 dysplasia is permanent. Perhaps it's sort of addicted to these tough cells. And along the same line of thought, perhaps the presence of these tough cells is promoting this chronic inflammation that we see in these lungs. However, there's also a possibility that there's an upside to this. Maybe if we have increased numbers of tough cells, this might be protective against certain kinds of pathogens that are typically cleared by type 2 immune response. So with that in mind, in collaboration with my neighbor, Dabrowski Herbert, we asked whether the presence of these tough cells in post-flu lungs might actually provide some protection against hookworm infection. And for those of you like me who knew nothing about the life cycle of these worms, essentially they get infected through the skin, they get into the bloodstream, they get caught in the capillaries of the lungs, where they then crawl out through the alveoli, up the airways, get swallowed, lay eggs in the gut. You can count the number of eggs that make it into the feces as a surrogate for sort of how productive the infection was. And the hypothesis here is that if we have more tough cells to begin with in the lung, essentially from 0 to many, that perhaps we can block these worms from completing their life cycle. And indeed, that seems to be the case. This was very reproducible across 3 different independent experimental cohorts. Now, of course, what we don't know at this point is whether this is really due only to tough cells or the myriad of other changes that are happening in these post-flu lungs. But after some struggle, I think we finally have a way to directly address that question, which is a way to deplete these tough cells after they have arisen. So we're using a tough cell specific constitutive Cree driver in this case, crossed with a Cree-activatable diphtheria toxin receptor. So we can then, after that, deliver diphtheria toxin intranasally, so straight to the lungs, essentially. And at least via the brush cell population in the trachea, which we kind of use to titrate the dose of diphtheria toxin, we can get very, very nice ablation. So we can go back to all those future directions that I asked and really be able to intimately pinpoint whether tough cells are responsible for any of these possible outcomes. So I think I'm out of time. So with that, I just want to thank my lab, former postdoc Nandy Costa, who unfortunately is no longer in the lab due to COVID-related travel restrictions and so forth. So we've kind of had to slow down on this project. Gargi, a technician, and I have been working on it in the meantime. And we just recently recruited a new postdoc, Marilena Gentile, who's taking over the work. I also need to quickly mention that my thanks for Noam Cohen, who I never would have thought to look at tough cells in the first place, had it not been for Noam's suggestion, and my frequent collaborator, Dabrowski Herbert. And also Mike Kotis from Richland. So very happy to take any questions. Thanks so much for the opportunity to speak.
Great, Andy. Thank you very much. That was a very interesting talk. Maybe if there's some time later on, we can have a discussion about whether tough cells are actually the right name, whether a tough cell is a tough cell is a. The next speaker is Rob Lee. Noam Cohen already mentioned Rob and the fact that he had done brilliant postdoctoral studies with him, but I should say that before that, he was a graduate student with me and did brilliant graduate student studies with me as well. So Rob now is going to present some work on taste receptors in immune cells. So Rob, take it away, please.
Okay, can everyone hear me? We're good?
Yep.
See my screen?
Yep.
Okay. So as Kevin said, I was a PhD student in his lab many, many years ago and learned a lot about live cell imaging, which I think comes through in the work that Noam and I did. And while in Noam's lab, I learned a lot about airway cell biology and in vitro culture models. And we're going to talk about some of that today. But I want to tell you something that's different than in the airway. We've heard a lot of airway stuff. So I'm going to talk to you about another project that's ongoing in my lab about taste receptors in immune cells. It's really nice that I don't have to present the background of taste. It's nice that I don't have to tell you that taste receptors are expressed everywhere, right? From your brain all the way down to your reproductive organs, and that we only know the tip of the iceberg about what these receptors do. And so a key question that all of our research and many other labs are addressing is, what do these extraoral T2Rs do? Beta receptors, also sweet receptors, T1Rs, and what activates them? That's a key question because we know some things about infection and bacterial metabolites, but there have to be hundreds, if not thousands, of endogenous ligands for these 25 different receptors that we just don't know. They're there for a reason, and I don't think it's just to detect bacteria. But a lot of the prior work that we've done that Noam talked about is looking at T2R38 and other T2Rs in cilia and their activation by acyl homoserine lactone gram-negative quorum sensing molecules. This activates a signal transduction cascade, which involves calcium, activation of nitric oxide synthase. In airway cells, it's the endothelial nitric oxide synthase isoform. This increases NO, which increases ciliary beating through protein kinase G. And it also activates bacterial killing by diffusing out into the airway surface liquid and killing bacteria. We've continued in my lab to work on a little bit of the various types of receptors that are expressed in airway cilia, including T2R14, which is one we identified in nasal cilia, as well as a little bit about their microanatomical locations. What's very fascinating about these receptors is they're not just cilia. They are, in the case of T2R4, the tip of the cilia. In the case of T2R14, more of the medial and distal cell body. So we're very interested in why the receptors traffic to cilia and what they do there. We've done a lot of work to define the signal transduction pathway, which, as I stated, involves calcium downstream of the G beta gamma subunit of the heterotrimeric G protein, which activates a calcium response that then activates nitric oxide synthase. What's really important and what is largely ignored is the other arm of the pathway. In taste cells, these receptors coupled to G-alpha-gasteucin, which Bob Margolski showed very importantly, that this couples to phosphodiesterase to decrease cyclic AMP. In many extraoral tissues, these receptors are coupled to GI G proteins, which decrease cyclic AMP, though, through inhibition of adenylyl cyclase. We're also really interested in another ignored part of these receptor signaling, which is kinase signaling, which we think is important for gene expression downstream of these receptors, as well as important for the nitric oxide synthase. The calcium we think is sufficient, but is necessary, but not sufficient for the NO production. So these are all things and pathways that we're interested in the lab. And I'm going to talk about these in a different cell type in a moment. One of the things that I think is really important to point out that we identified a couple of years ago is that it's not just acyl homoserine lactones that activate these receptors. There are also these compounds secreted by Pseudomonas and other gram-negative bacteria called quinolones. And they look remarkably, this is Pseudomonas quinolone signal, this is heptyl hydroxyquinolone, and this is dihydroxyquinolone. They look slightly like quinine and chloroquine. So just based on that, we tried it in heterologous expression in hex cells expressing different T2Rs, identified different T2Rs that respond to these molecules, including T2R4, 16, 38, and 14 that are expressed in cilia. And we've also validated that these molecules activate these innate immune responses in primary epithelial cells, similar to classic bitter taste receptor agonists like quinine. Another aspect of T2R signaling that we've been interested in is plant compounds that activate T2Rs. So a lot of, almost everything we know about bitter agonists come from plants. And some of these plant compounds, so Ben Hariri from my lab worked on these compounds called flavones, which activate T2r14, which is expressed in cilia. And these plant flavones actually can activate T2R. So one important aspect is not only are these receptors, do we think, immune sentinels, but we think that understanding the biology may be really important because T2Rs may underlie some effects of homeopathic therapies, right? If we can understand what the molecular basis is for some of these effects, maybe we can actually optimize them and come up with new ways to treat airway diseases based on plant products that people have been using for centuries that are maybe, have completely unknown mechanisms of action. So I want to talk to you about endogenous T2R signaling outside of the airway epithelium because you've heard a lot about the airways. So one of the things that we're also interested in the lab is using our tools and expertise to study endogenous T2R signaling in other cells and other tissues. And one of our concerns in the lab is that evolutionarily orthologous mouse and human T2Rs aren't necessarily activated by the same agonist. So while mice are absolutely required for organismal physiology, we want to stick as much as we can with primary human cells so that we can understand if and how the human T2Rs we discover in different cell types are actually druggable. And we wanted to stick with cell types where the idea of bacterial ligands makes sense, where the cells are hopefully serving an immune role. To extrapolate from that, other labs have shown that several types of immune cells express T2Rs, including neutrophils. Most circulating leukocytes express a subset of bitter taste receptors, as well as a subset of olfactory receptors. And while these receptors have been characterized on expression, very little is known about the function of these receptors in these cells. I am going to talk today about T2R signaling and macrophages, which is where we started. So the reason we started looking at T2Rs and macrophages, there's a scientific reason and a logistical reason. I'll start with the scientific reason. The first is that they're early players in innate immunity. These are things that need to be armed to respond to whatever pathogen they encounter. They're not already programmed. They have to be generalists. They have to be able to respond to bacteria fairly quickly. And as I mentioned before, other people have shown that macrophages express T2Rs, but absolutely nothing is really known about signaling or function. And what also is really interesting about macrophages is that they don't have motile or primary cilia, where almost every cell type in your body has a primary or a single sensory cilia at some point in development. The cells in your airway, the cells in fallopian tube have motile cilia. Macrophages are one of the cell types in your body where we've never observed any type of cilia at any point during differentiation. And so that is really interesting because we know that T2Rs like to traffic to cilia. So if they're expressed in these other cells that have no cilia, where do they go? What do they do? Logistically, macrophages are really a good starting point because we can get tons of human monocytes from healthy apheresis donors from the UPenn Human Immunology Core, which has been just a wealth of supply of cells from which we can do a lot of great experiments. We can take human monocytes that come out of patients, we can differentiate them into macrophages over 10 days with a defined cytokine mix. We can then even further differentiate them if we want into M1 inflammatory macrophages or M2 tissue repair macrophages using defined cytokines. And macrophages in vivo probably exist in a gray level in between some of these extremes. But these are just models that we can use in classical ways to polarize macrophages that we can look at how taste receptor signaling expression changes. The data I'm going to talk to you about today are entirely in M0 or unprimed macrophages. So these are not that indistinguishable from monocytes. There are markers that differentiate them, but they are sort of the most naive variant of a macrophage that you can get. So lo and behold, what we saw is that the receptors, at least several receptors, including T2r46 and T2r4, T2r4 is one of the receptors that detects Pseudomonas quinolones, traffic really well to the plasma membrane in macrophages. We get really nice membrane staining, a lot like what we get with GLUT1 shown here as a control. We can see the receptor expression via just simple RT-PCR. And we can look at the differential expression with quantitative PCR, and we can see that some taste receptors are expressed really well. Some taste receptors are not expressed at any detectable level. One of the most notable is T2r14, which many other labs have shown is one of the highest expressing T2Rs in extraoral tissues, including T2r4, including T2r46. So, and what's also really interesting is there's not a whole lot of difference between M0 polarized macrophages or once we polarize them to an M1 or an inflammatory phenotype with LPS and interferon gamma. And M2 polarization is actually a little different. I don't actually want to show the data right now because we're still trying to figure out some of the mechanisms, but we think that taste receptor expression actually goes up with M2 polarization. And we think that that's where this signaling pathway may be even more important or may do other things. So we take macrophages, we load them up with a calcium sensitive dye, Fura2, and we stimulate them with a classic bitter agonist, quinine. This is what Noam is really interested in putting up people's noses as a therapeutic. We can see an increase in intracellular calcium. This is a very small increase relative to what we get with purinergic agonist ATP. And this is a hallmark of these receptors, are small intracellular calcium increases in extraoral cells that we've looked at. Pseudomonas quinolones also give us small intracellular calcium responses. We can look at, if we look at T2r14 specific bitter agonists, we can knock down this response with T2r14 siRNA. We think this is specific for the receptors. And we've demonstrated that they're T2r receptors, they couple to calcium. This is a phospholipase C dependent pathway. We've also shown that the other arm of the pathway, the decrease in cyclic AMP, is also there. If we transduce these macrophages with a fluorescent protein biosensor, this is something called Caddis from a company called Montaigne Molecular, and their BACMAMs, actually, we've been really successful with transducing macrophages. If we stimulate macrophages with Pseudomonas C12HSL, this acyl homoserine lactone T2R agonist, we get a decrease in cyclic AMP. If we stimulate with forskolin, which is a adenylyl cyclase activator, we get an increase. And if we treat the cells with pertussis toxin, which kills GI-coupled G protein signaling, both arms, we can block that effect. What's also interesting about macrophages, which we didn't even know when we started the project, is that they also, M0 macrophages express a good deal of nNOS and a little bit of eNOS. And so we wanted to know, do these receptors couple to nitric oxide synthase production the same way as they do in the airway? Lo and behold, we loaded macrophages up with our nitric oxide sensitive dye, DAF-FM. We saw really nice increases in NO with bitter agonist stimulation. If we treated the cells with pertussis toxin, there was no NO production, suggesting this is downstream of these bitter taste GPCRs. And we can also measure cyclic GMP production via a similar type of fluorescent biosensor. If we stimulate with bacterial or other T2R agonists, we get an increase in cyclic GMP. Pertussis toxin and inhibition of nitric oxide synthase knocks this out. So we've defined that this arm of the pathway, which is really interesting, in airway cells also exists in these unprimed macrophages. So the question is, what does it do? So what do macrophages do? They eat things. We thought that it was very appropriate for a study of bitter taste receptors to begin to look at how the macrophages ate things. And lo and behold, we found that when we stimulated our macrophages with bitter taste agonists, they eat about 3 times as much FITC labeled E. coli as they do when they're unstimulated. And we've confirmed that this is phagocytosis with pH sensitive dyes labeled on bacteria. And what's interesting is that this is really dependent upon this nitric oxide synthase response. So activate the macrophages with a bitter taste receptor, and over the course of 15 minutes, they'll eat a lot more bacteria. If you inhibit nitric oxide synthase with this compound L name, but not the inactive compound D name, you can block this effect. You can scavenge nitric oxide synthase and nitric oxide with carboxy-PTIO. You can inhibit protein kinase G with KT5823, or you can knock out the G protein signaling. So this pathway is really critical for stimulating phagocytosis acutely within 15 minutes, we think, when macrophages encounter these bacterial metabolites. This is essentially our model that we get T2r signaling. We can get the same responses, but they do a very different job. They do a very different physiological output. And what I don't really have time to show you is that through co-culture methods of different cell lines as well as primary cells, what we found is that the NO produced by airway cells is also a signal that is sufficient to boost macrophage phagocytosis. So we think spraying these bitter compounds up someone's nose, which is going to stimulate airway cells to produce NO, may also have a beneficial effect to stimulate immune cells to come in and clear out infection or clear out viruses even better. So you may be, and one reason why these receptors may be so important for airway physiology is that they're not just having effects on epithelial cells, they're having effects on immune cells as well. And so some key points that I think the whole symposium covers, but from my talk, is that really these T2Rs are taste receptors, but they're also immune receptors, right? They're chemosensory receptors that are used by our bodies for multiple things. In airway cell cilia, the calcium response activates NO production, you get ciliary beating, you get direct bactericidal effects. In macrophage plasma membrane, you get calcium, you get NO production, you get increased phagocytosis. A paper was just published that's shown that NO production downstream of taste receptors inhibits hormone secretion by ovarian cells. So there's a lot more going on here. We think the genetics of taste may underlie susceptibility to infection beyond chronic sinusitis. And we think that extraoral T2Rs may contribute to off-target drug effects, efficacy of plant-based therapies, and we're working to better understand the GPCR biology to discover how we can leverage the T2Rs to treat infections, what other processes the T2Rs might regulate. So just to acknowledge really quickly, my lab, our collaborators, Penn Rhinology, particularly Noam, who got me started off on taste receptors, including Danielle Reed and Bob Margolski, who got Noam started off on taste receptors many, many years ago. I really like to thank the Penn Immunology Corps, who've been very great at providing cells. Indy Gopalawa has done some of the macrophage work that I showed, as well as Jenna Freund. Jenna Freund also did some of the PQS work, and Ben Hariri did a lot of the quinoline and plant flavone work. So thank you very much.
Thank you, Rob. That was wonderful. I know I see a lot of questions. I have a lot of questions, but it is time for our coffee break. So I would invite those of you who need to slip off and get your coffee and whatever, please do that. Those of you who want to hang around, I have questions to ask Rob. Hopefully he'll answer them informally. And then at 11 o'clock, so in a few moments, then we are going to be talking about some of the real questions in chemosensory biology, and immunology is really focused on post-viral smell loss, including COVID-19. So we have two fantastic speakers lined up for you starting at 11 o'clock, followed by a keynote address by Nancy Rawson. So
good morning. Still morning, I think. Very important morning. Thank you, Danny, for that introduction. Actually, I think if anyone has been paying attention recently to the popular press, it's become pretty obvious that smell loss has taken front seat in terms of stories about COVID-19. As it turns out, it's one of the cardinal symptoms of infections. Now, as Danny said, olfactory dysfunction secondary to an upper respiratory infection is a well-recognized and actually a pretty well-studied phenomenon prior to COVID-19, but probably at no other time in history has it been shown to occur with such a high prevalence and especially affecting so many people worldwide simultaneously. So the goal of my talk today is really to set the stage for my colleague Jay Gottfried's talk by giving some background on what is known about post-viral smell loss pre-COVID-19. So there are two ways that smell loss post-virally has been classified, quantitatively and qualitatively. Quantitative loss can either be complete, which is anosmia, a complete inability to smell, or hyposmia, which can be various grades of smell loss, resulting in a change in olfactory threshold and still some residual ability to smell. There are also qualitative changes that can occur as a consequence of post-viral olfactory disorders. The most prevalent is parosmia, which is a distortion in the percept of what is being smelled. Generally, it is a familiar odor that no longer smells like what you expect it to, and unfortunately is often a very unpleasant percept. Phantosmia is less well reported, but it can still occur as a consequence of post-viral olfactory disorders. Phantosmia, in contrast to parosmia, is the perception of an odor, again, usually unpleasant, but in the absence of any source. Well, from a pathophysiological perspective, olfactory dysfunction generally and post-virally is usually classified in two ways. A conductive loss, which really means an ability or an inability of the odorant molecules to find their way to the olfactory epithelium and bind with odorant receptors. And this can occur as a consequence of different kinds of disorders such as congestion, mucosal edema post-virally. It's also very common in nasal sinus disease, in people with polyps, and other kinds of inflammation in the nasal cavities. In contrast to conductive disorders, there can also be changes in the sensory neural apparatus of the olfactory system. And this occurs certainly after upper respiratory infections, but also in neurodegenerative disorders and sometimes Kalman syndrome like congenital disorders and toxin exposure. The reason that these disorders are classified this way is somewhat paradoxical, because in fact, they can both occur at the same time. What I mean by that is you can have a conductive disorder that will resolve, but still have a sensory neural disorder that will result in a persistent loss. So what are some of the most common etiologies of acquired smell dysfunction, just to put post-viral in context? It turns out that nasal sinus disease affects more people and results in more disruptions to the olfactory ability. But upper respiratory viral infections are only secondary to that. And so it is very common following an upper respiratory viral infection to have either an acute or some cases a persistent loss of smell. Head trauma and toxin exposure result in some degree of olfactory dysfunction, but much less. Now, it turns out that upper respiratory viral infections are also actually indicated by not only resulting in a loss of sensitivity, but may also be more likely than these other disorders or these other etiologies to produce parosmia, which is a change in the percept of what is being smelled despite the same source. So when we look at a viral upper respiratory infection, it's very common in the early stages that there will be a conductive or what we would call an obstructive loss secondary to swelling in the mucosal areas or other types of congestion. Fortunately, that kind of disorder generally, if it's the only one that's being affected, actually leads to fairly good recovery, which is why most people who experience many viral URIs over the course of their lifetime will never actually experience a persistent loss of smell. However, there can be, as well with conductive disruptions, a change to the sensory neural area as well. And this is where we get into more persistent or permanent post-viral olfactory disruptions, which can result in either complete anosmia, hyposmia, or the condition I described as parosmia. Under some circumstances, those individuals will go on to recover either fully or partially, and sometimes that recovery can take up to 2 to 3 years, but it does occur. Unfortunately, in a percentage of those individuals, they will be remaining with a persistent or permanent post-viral olfactory disorder. And upper respiratory viral infection is the most frequent cause of persistent sensory neural olfactory disruption. Now, PVOD can be caused by disruption at multiple levels of the olfactory pathway. So, for example, conductive disruption is not always present, but it can be. In this case, PVOD can be attributed to direct neuronal damage, inflammation, or even effects of inflammation by cytokine presence in the olfactory cleft. And here I'm looking at a study that was done by my colleague and I, Hong Wang, where we found that the prevalence of a variety of inflammatory mediators in the olfactory cleft was negatively associated with olfactory performance. It's also the case, and this is something that is really in the beginning of its ability to identify this, but the degree and locus of dysfunction does appear to be virus specific. So in a recent review that was done of both animal and human studies, influenza virus was shown to produce lesion changes in the olfactory epithelium and the olfactory bulb, whereas SARS-CoV-1 did not show the same level of gross lesioning in the OE, but did impact the turnover of olfactory receptor neurons and also affected high cortical regions. In human studies where the virus was unspecified, post-viral effects were found to cause degeneration and death of olfactory receptor neurons, as well as reduction in olfactory bulb volume. And this occurs in the absence of conductive disruption to the system. So in another study where they were actually able to identify from nasal secretions the type of virus that was affecting people with post-viral olfactory disorder, they found that even after 4 weeks, mucosal cross-sectional area, which is a proxy of a measure for congestion, actually improved dramatically in these patients, but their olfactory disruption did not improve. And so most commonly, they were identified as rhinoviruses and to some degree, some coronaviruses, parainfluenza virus, and Epstein-Barr virus. But even when the conductive changes and congestion resolves itself, there is still evidence of long-term effect to the olfactory region. Okay. So this was a study that was done in 2006 by Rombaugh showing that the olfactory bulb volume is reduced among patients with post-viral olfactory disruption, and the volume is related to the duration of olfactory disruption. On the left here, we have someone, essentially a normal MRI image of a non-infected olfactory tract and bulb. And on the right is an MRI image of an individual with post-viral olfactory hyposmia, where you can see that the olfactory bulbs are actually quite reduced in volume. Let me go back one second. In this case, this individual also was aware of having a parosmic episode. So, and that seems to also be associated with changes in the volume of the olfactory bulb. It's also the case that the longer the disruption has occurred to the olfactory system, the smaller the volume of the olfactory bulb. And so interestingly, if you look at these two distinct causes of olfactory disruption, post-viral and nasal sinus disease, a recent study showed that actually post-viral olfactory loss is more disruptive to mood. These are Beck depression inventory scores from people, and they equated these individuals in both groups on the degree of hyposmia that they were experiencing, or anosmia. So their degree of olfactory loss was very similar, but the individuals who had experienced post-viral URI were much more affected in terms of their depression scores than people with nasal sinus disease. Now, that may be related to the fact that post-viral olfactory disorder results in more parosmia than does nasal sinus disease. It also may be the case that nasal sinus disease actually occurs over a longer period of time in producing olfactory disruption, whereas post-viral olfactory disruption is generally a very acute onset following the time course of the virus. But here we can see that parosmia is a pretty common symptom of post-viral olfactory disruption. This was a review done by Steve Nordin and his colleagues some time ago, looking at studies and clinical data from both the US, from Europe, and Asia as well. And basically what they showed is that the complaints of parosmia were significantly higher in people suffering from post-upper respiratory infections than they were in people who had lost olfaction due to head trauma or nasal sinus disease. And if you have the opportunity to go to any of the Facebook groups of a place like Absent, for example, or anybody reporting parosmia, you will find that they find it often is a far more distressing condition where things that you expect to smell a certain way no longer do and generally smell very unpleasant. And this is probably a more distressing condition than simply not being able to smell at all. So what can be done? Well, another recent review looking at treatments of post-viral olfactory dysfunction looked at various classes of treatments, starting with systemic steroids, which they categorized in terms of both the harm-benefit ratio and whether these could be recommended as options. Systemic steroids, they felt that the harm and the benefit were relatively equal. And so therefore, as a short-term course in the early stages of post-viral olfactory loss could be recommended as an option. Topical steroids had the same profile in terms of their harm-benefit ratio and also were recommended as an option. Similarly, for non-steroidal topical treatments, the one not recommended treatment was zinc sulfate because of the potential harm that they felt that outweighed the benefit. And then there were a number of studies looking at non-steroidal oral medications such as antibiotics, theophylline, vitamin A, and alpha-lipoic acid. Under those circumstances, while the harm did not outweigh the benefit, they came away with the impression that there was no recommendation to be made. And that was true as well for traditional Chinese acupuncture. However, olfactory training, of which there were 10 studies reviewed, showed greater benefit and certainly very little harm other than a degree of effort on the part of the person who was trying to recover olfactory sensitivity. And this was highly recommended. Now, olfactory training can take various forms, but it generally relies on choosing or being given a number of common everyday odors to smell, and in some cases relating them to the name of the odor, the source of the odor, the identity of the odor. Now, whether the benefit is a result of frequent olfactory stimulation or making a connection between the peripheral signals and the corresponding readout in the bulb or the cortex, it has definitely been shown to be helpful in restoring some degree of olfactory function in these cohorts. So just to summarize, olfactory dysfunction of various durations occurs in between 18 to 45% of patients following an upper respiratory viral infection. Anosmia and hyposmia are very common symptoms of post-viral olfactory disorder. Parosmia is also more common among post-viral patients and may be related to the process of recovery or axonal regeneration from the olfactory epithelium to the bulb. post-viral olfactory disorder is also associated with higher scores on a depression inventory than the loss of smell from nasal sinus disease. And currently, olfactory training is the only treatment shown to be somewhat efficacious and recommended under these conditions. And with that, I thank you very much.
Pam Dalton, thank you very much for your talk. I direct everybody to put their questions in the chat, and Pam will be happy to deal with them there. And I will turn this over to Kevin Foskett to introduce our next speaker.
Yes, thank you, Danny. Our next speaker is Jay Gottfried, the Arthur Rubenstein University Professor of Psychology at Penn. So Jay, if you're ready to go ahead.
All right, let me give this a try. Okay, is this all visible, Kevin?
Yes, it looks good.
Great. Well, for starters, I sort of feel like I've sneaked into this really amazing symposium as one of the lone smell people, along with Pam on this. But anyhow, I think there's been a lot of really great complementary research and ideas being presented here. So I'm very proud to have been asked to take part. So this is just my banner here for the COVID talk. On the left is the virtuous inflammatory cells trying to eradicate our virus particles to the right. Anyhow, so what I'd like to do is provide a very brief overview of the human olfactory system, and then from there move into the realm of COVID. And I thank Pam up front for helping set up some of what I'll be discussing. So this is a cross-section through the half brain. What we're looking at here is the lateral turbinates along the right part of the head, where the remainder of the skull has been removed. And the idea here is that, let's take, well, one of Philadelphia's favorite foods, the Philly cheesesteak. All these aromas that emerge from this cheesesteak travel up into the upper reaches of the nasal epithelium, where they activate the dendritic endings that are poking right into the nasal mucosa. And from there, that information will be trafficked to the bulb and points further downstream. So one really key thing to take away here is what I've circled in light blue is central nervous system tissue. So basically, we have neuronal tissue poking out into the nasal cavity. So it's not hard to imagine how many different kinds of particles and components would have very direct and easy access into the brain through the olfactory route. And I just love showing these slides whenever I can. These are freeze fracture electron micrographs at different stages of magnification. And what you can see labeled O, these are the olfactory sensory neurons with their stalks coming up to the surface, forming this really dense mat of olfactory receptor endings, amplifying the ability to detect odors at very low concentration. Labeled in SU, this is a supporting cell, or what's often called a sustentacular cell. And this does all of the hard work in supporting the functionality of the olfactory sensory neurons. And in this really blown up micrograph, you can see kind of the beauty of this ciliary tuft in the olfactory mucosa, resembling a sea anemone. So very briefly, The anatomy of the olfactory system in humans is very unique. We have our two olfactory bulbs looking like earthworms run over after rain. These are the two long, skinny tracts that end up terminating into different areas of the medial temporal lobe. So from the nose, these chemicals are taken up by the olfactory sensory neurons, where the signal is passed downstream to the olfactory bulb. And this projects to many different cortical regions in the brain, which, to put simply, are involved in various aspects of interpreting and encoding questions about the what, why, and where of an incoming smell. And all of this information gets kind of bundled together in an integrated way in the orbitofrontal cortex. So one important message to get across here is that the olfactory cortex is extremely well connected. So this is a rodent study that was done some time ago, which nicely illustrates the point. If you take an individual piriform olfactory neuron in the brain and inject it with a tracer dye, you can see that this one cell is able to speak with many other different regions of the brain, most of which are critically involved in different aspects of regulating emotion and behavior. So a lot of the brain listens to what the olfactory system has to say. Now, the clinical relevance of smell, which will kind of dovetail into my COVID discussion here, it's clear that the olfactory system is susceptible to many different forms of neuropathology, whether this is epilepsy, traumatic brain injury, neurodegenerative disease, especially Alzheimer's and Parkinson's, and of course, Pam touched on some of this. So one message I'd like to convey here is that the link between smell and neurological disease specifically offers a unique opportunity for understanding how to diagnose these disorders at the earliest stages and how to prevent these disorders from gaining traction in the brain. So let's turn to COVID-19 and smell loss. I did a little bit of armchair research looking through PubMed, and as of just a few days ago, there have been 753 articles published specifically on the topic of COVID-19 and smell loss, which is really quite remarkable. And this spans all different areas and domains: epidemiological studies, otolaryngological, neurological, pediatric, pregnancy and COVID, and it's had a truly global impact, likes of which I think we've seen for a long time. And I'm not going to read through all of these countries that have been involved, but you got to like the fact that even the Democratic Republic of the Congo has participated in helping us understand mechanisms and epidemiology of COVID-19 and smell loss. So I'll quickly go through this slide, as Pam has said a lot of it already. Olfactory dysfunction is prevalent. Post-viral causes are a dominant etiology. Very briefly, if you ask people how their sense of smell is over age 40, up to 10% of people report that they have some dysfunction in their ability to smell. With objective testing, these numbers increase with age. There's also functional anosmia, which is kind of reported consistently at a low baseline of about 5%. And olfactory dysfunction increases with age, with post-viral being a leading cause, along with traumatic, nasal and paranasal disease, and then other idiopathic unknown etiologies, likely post-viral. Some trends that have been shown over the years in terms of post-viral olfactory insults There is a lot of inter-individual variation from one person to the next. Olfactory bulb atrophy can be identified on MRI scans, and there's a very atypical cell morphology and tissue landscape when you conduct biopsies. Here you can see on the left a very rich and olfactory sensory neuron full epithelium with a very wide span of epithelium as seen here. But following a post-viral insult, you can see that on the right, there's a lot of disruption in the cellular features here, and arguably there's very little remaining classic olfactory type epithelium here. So in terms of COVID-19, we've learned a lot since this all started. I would say that everything is still in a preliminary state. That is, there are still many more questions than answers. Generally speaking, the prevalence of hyposmia, which is reduced smell, and osmia, which is complete smell loss in COVID-19 patients, those who self-report, the numbers appear to be roughly 60% with a very wide range. With objective testing, the range is a bit tighter and also quite high. Some key points here. It can often occur, that is smell loss with COVID, can often occur in the absence of nasal obstruction and rhinorrhea, which is very different from a more classical kind of viral insult that would be seen in more conventional viral syndromes. Smell loss can be the sole symptom. It can occur before, during, or after other COVID-19 symptoms. And the recovery rate is generally thought to be faster than post-viral smell loss from other viruses, and as many as 75% of individuals do recover over 4 weeks, which is a very different kind of clinical trajectory than what is seen with other viruses. And just to show one example here, there have been a lot of really amazing crowd-based research studies being conducted. This one primarily from the UK, using a phone app, recruited more than 2.5 million responders, which is just really incredible and has helped us learn a lot more about the epidemiology of smell loss and COVID. And they've even kind of developed a predictive model to assess what are the features by which COVID-19 is more likely to lead to smell loss. So as for mechanisms, there's been a real flurry of research being done across numerous different labs, many labs across the US doing great work. So I'd start here by saying that many patients report anosmia without rhinorrhea or nasal congestion, as I previously noted, and olfactory bulb imaging is either normal or reveals focal olfactory bulb inflammation. Again, this is very different from the more conventional viral insults that are seen. So this is a fun MRI scan. What you can see in panels C and D are coronal sections, imaging sections of the olfactory bulb and adjacent tissues. At day 7, this patient with anosmia has what you can see in kind of a bean brown color, very plump looking olfactory bulbs reflecting inflammation. If you look to the right, 24 days after infection, these bulbs have really come back to their more typical size. So there's a very dynamic change that takes place in response to COVID in the nose. And what is rewarding to see is that as I mentioned before, many of these patients do regain their sense of smell. So, along these lines, an important point is that when COVID-19 attacks the olfactory system, it appears to be using mechanisms very distinct from those used by the more benign endemic coronaviruses. And some of this information here is from a really great and thorough review article that came out a couple months ago by Cooper, Brand, and colleagues. So in vivo animal models suggest that COVID-19 infects supporting cells, and I showed you those supporting cells from before. These are almost like providing a Trojan horse entry into the olfactory system and brain. The olfactory sensory neurons themselves don't seem to appear to be directly a target of infection. Infection requires co-expression of ACE2 and the TMPRSS2 protease and or other proteases, which results in cleavage of those S-spike proteins you've seen in those really pretty coronavirus cartoons, if only they were as kind as they were pretty. So a key point is that ACE2 itself does not seem to be expressed in the olfactory neurons, but only in the supporting cells, such as sustentacular cells and Bowman glands. Current data suggests it is unlikely that COVID-19 directly infects neurons in the bulb or directly conveys the virus to the cerebral cortex. Rather, secondary vascular effects and inflammation are more likely responsible. I'm getting to the finish line there, Kevin. A few studies suggest that COVID-19 is neuroinvasive, but these reports at the moment are really very preliminary. So I showed you this before with the sustentacular cells and the olfactory neurons. Here's my little movie cartoon. So if we orient everything here, here comes the COVID bug, which can't get through the olfactory system pathway because it doesn't have the right carriers, but it can do so through the sustentacular cells and indirectly infiltrate. This is just a really pretty, colorful, and informative slide showing that really, in terms of neurological complications, it's systemic inflammatory responses, prothrombotic clotting states, and viral infection, which together create a lot of inflammation and edema, leading to the changes that are being seen. And this is associated with obviously with a stroke and other vascular pathology throughout the body. And this is a nice quote, but I won't read it for sake of time. It pretty much says what I've just pointed out, apart that it seems that the microvascular disease and hemorrhages, along with coagulopathy, are really key here in terms of what's going on at a neuropathological level. I'll move quickly through this. We're running our own study based on subjects who have presented to the Penn testing site. We have made an effort to try and correlate the degree of smell loss in individual subjects to the severity of their eventual disease, to develop a kind of predictive model of who might need to be prioritized for more aggressive care. And I'll just move here towards the bottom. We've recruited 646 patients, 528 have completed testing using a 12-item smell test at days 0, 4, 12, and 60. Our COVID positivity rate is 8.35%, and we're just about done with data collection, and we'll soon be running the stats to see how this all maps out. So I'll end here. Thank you for your attention. I just highlight Kyle Mabry, Clara Reitel, Naz, Dikesh Ligil, Sepideh Chehavtani, Lulu Korsak and Sarah Kwan, who have been really instrumental in that study. So thank you very much, and I'm happy to take questions.
Thank you, Jay, very much. So I would direct the audience to put questions for Jay in the chat. Very interesting new data. I'll be very keen to see when you're further along in the analysis there. So I'd like to introduce our next and final speaker. So as you know, this symposium has been a joint effort between the University of Pennsylvania and the Monell Chemical Senses Center, and I think we've seen the fruits that combining chemosensory biology and immunology can bear. And so I'd like to just introduce our Vice President, Nancy Rawson, who's going to give us her vision of how we might work together more effectively in future. So Nancy, go ahead.
Can you hear me okay?
I can hear you and we can see your slide, but you're still in presentation. You need to put yourself into presenting mode.
Yep. There we go. Is that better?
You're in.
Wait a minute.
Yep. Yeah.
Yep. Field swap. So thank you, really, Jay, for putting up that picture of the Philadelphia cheesesteak right before lunch, so that we can all anticipate lunch here. And I'm also mindful of the little event happening at noon today. So my remarks will be relatively brief, but I really do appreciate the opportunity to talk a little bit about looking ahead and in thinking about what I wanted to say, I came across this: if you want to know the future, look at the past. And I thought that it really would be worth giving a little nod out to some early work that was done at Monell by our Director Emeritus Gary Beecham and colleague Kunio Yamazaki. And this is from a paper in 1981 where they looked at they showed that distinctive urinary odors are governed by the major histocompatibility locus of the mouse. And this really was the beginning at Monell of this interest in connecting immunology and the chemical senses. Gary later went on to say that genes known to be critically involved in regulating immune responses also control olfactory individuality, and a link between immunology and olfactory communication has been established. And I think the work that we heard from Bruce kind of picked up on this idea and has carried that forward in really, really exciting ways. To tell you a little bit about Monell today, our mission is to improve health and well-being by advancing the scientific understanding of taste, smell, and related senses. And we have outlined 4 strategic aims, research aims. And what I will do in the next few minutes, is to kind of connect these aims with some of the work that has been presented and what I see some really exciting opportunities moving forward. I also looked at the strategic plan from Penn Medicine and was really excited because there's a lot of important goals and goals that I think really we can connect to through the work that was presented today. So one of the areas was to prevent disease before it occurs, and when disease does occur, to diagnose it early and cure it. And so the work that Danny and Noam are doing on taste receptor genetics and sensitivity to respiratory infection and treatment response really is just so exciting, because this is not a distant future that we're talking about. This is really happening as we're speaking, that we can develop individualized treatments strategies for sinonasal disease. So this is really a nice model for how this synergy can really advance progress in some of our very important health challenges. We also heard Bruce talk about chemosensory signatures reflective of disease states, and the advances made in that in many different areas that really, by working together, I believe that we can truly diagnose disease early before other symptoms using these volatile metabolomic signatures. In some other areas, we heard about TAS2R expressing solitary chemosensory cells in the oral cavity and resistance to periodontal disease from Marco Tizzano. We heard about how tough cells influence GI health, and the really exciting work by Rob on TAS2R expressing immune cells. Perhaps the immune system isn't similar to the chemosensory system. Perhaps it is a chemosensory system. And a lot of what we heard about relates to the microbiome and the microbial environment within all of our tissues. So I think that this work really is leading to prevention and personalized treatments for periodontal disease, GI infections, and really a lot of other serious health conditions that we're all facing today. Another important area for Monell is in tackling the loss of smell and taste. And one of the also themes at Penn is to invest in fundamental science that can have the greatest impact, and leveraging new findings to test in early treatments. We've heard about how loss of smell and taste is a growing concern, and currently really there's minimal treatment options. Whether you're talking about COVID-19 or aging or neurodegenerative diseases or other immune system types of disorders, as Hong spoke about, this condition is really quite serious and has very little treatment opportunities. And we also heard about immune modulation of taste and smell function and regeneration processes. And one of my questions is really, I think that these things can be used for new approaches for the prevention and treatment of chemosensory disorders. We also heard about the immune response and taste cell survival and regeneration. One of my questions is, how do these factors impact on dietary habits and digestive function? And I think that's the next area, the next frontier that we need to begin to address. But I think if we can, it may lead to new approaches to treat obesity, metabolic, and GI disorders. And finally, going back to taste and smell loss, I wanted to also highlight the fact that there's now a number of patient groups, the Society, the Smell and Taste Association of North America, Absent, and another number of others that are really drawing attention to the importance of this area. And if we can develop ways to ensure that our senses of smell and taste will be healthy throughout life, I think we can improve the nutritional status that is affected in so many of these serious health conditions that the world is facing today. So what's next? I hope that today's symposium will stimulate a lot of discussion amongst the scientists, amongst the organizations to really streamline and develop new ways of working that are getting us to treatments and new options really quickly, that will come up with a lot of great ideas. Of course, the way to have a great idea is to have lots of ideas. And I want to thank the speakers, and I especially want to thank our hosts and our organizers. I thought the whole morning was just stupendous, really exciting, really informative, and thank you all, and enjoy the rest of your day.
Thank you, Nancy Rawson. That's very inspirational. So that just leaves Kevin and I to thank all the speakers, and for the audience members, I know we're all eager to log on for our upcoming national event. So this will end our formal presentation and our formal symposium. So I wish you all well. Thank you. Bye-bye.